Good morning, everybody. Thank you so much for agreeing to participate in this effort. We are doing this as one of our workshops without walls, which is a tradition here at NAI. We think we invented it, and we have done it for a huge variety of topics and with great results. So let me give you my own personal perspective on why astrobiology, why now. I am not a virologist, although I appreciate viruses, and I think I'm seeing them everywhere in my own work. But I hold the position that the late Dr. Barry Blumberg held, and of course he was a Nobel laureate winning virologist and chemist, and who worked extensively, of course, with hepatitis and various other aspects of medical virology, but who was, of course, extremely passionate about astrobiology and was the first astrobiology director of the institute I now head. We are in our 20th year, and NAI is going away as an entity and being replaced by a new way of organizing astrobiology within NASA. But I have always felt, since Barry passed away, and he was a good friend of astrobiology and a good friend of mine, and at the time he passed away, we were in the process of sort of pre-organizing an effort to do a field trip for the astrovirology focus group into caves to actually look at what the virome situation might be in cave environments, which is where I typically work and do primarily bacteriological and archaeal studies. So when Barry passed away, of course, the plans for that halted. And so before we sunset the Institute, I would like to do something to honor his great work in that field and his passionate devotion to astrobiology and his faith in pursuing the astrovirological aspects of astrobiology, which I think are extremely important and have not received the attention that many other areas of astrobiology have. On top of that, of course, why now? Part of the reason is because for those of you not familiar with the NASA and its processes, one of the things that governs what we choose to do in science and missions is a every 10-year so-called decadal study that happens within all of the 4 major science branches of NASA. So NASA is comprised of a heliophysics branch, an astrophysics branch, an earth sciences branch, and a planetary sciences branch. And astrobiology is housed currently within the planetary sciences branch. Well, it so happens that we're coming up on the next decadal survey, which is conducted as an external advisory and assessment activity by the National Academy of Sciences. So the NRC, or whatever it's calling itself this week, the academies are the entity that actually does this assessment and produces a large and very attention-getting document. And by attention-getting, I mean that Congress pays attention to what the guidance is that we receive from the national academies. And so one of the ways, in fact, one of the main ways to get attention for a particular field or a particular type of mission concept or whatever it might be that one is pitching is to produce a white paper for that process. Now, the latest intel I have about this is that the letter of authorization from NASA has not yet been received by the National Academy, so we don't know exactly the precise timing, but word on the street has it that the white paper submission process will be opening in February and that it will close probably in the April to May timeframe. And then, of course, the panels that are constituted as part of the decadal process will then deliberate. And so it is very timely for us to engage in this now. This the middle of September. that gives us October, November, December, January, February, to about the middle of February. So that's 5 months to really try to see whether we can produce a consensus white paper. The good thing about the white papers is that they're very short. The bad thing about the white papers is that they're very short, and so it's very challenging to write them. One of the things that is really important about white papers is that instead of a particular field or mission concept, idea, or planetary body, for that matter, having a zillion different papers, all with a few authors on them, what really has the most impact is a limited number of papers with a very large number of co-authors and co-signers. Or we make everybody a co-signer, or we do whatever we want. There's no good template for writing these, so they are all over the map. The astrophysics ones that just came in, because astrophysics is about a year in advance of the planetary, and so that's already well underway. There were 940 white papers, which is probably not a good thing that there were that many. And many of them had, one or two authors, not very ideal. And of course, their formats were all over the place. And so that means that you can't show a groundswell. I think with the, relatively small and intimate nature of the astrovirology community, of which I am not part exactly, but I'm a fangirl and trying to promote, that I think that we ought to be able to come up with one or at worst two documents that really promote aspects of astrovirology that we believe are germane to NASA's mission to understand the origins and evolution of life on Earth, and to extend that to other planets, and to seek evidence of life elsewhere. And then one of the third components that is often left out of the picture is the aspect of what is the future destination of life on our planet and beyond. So really the big questions that NASA has scoped out are really all across this time spectrum and broadly across the life sciences as they apply to space and Earth's deep time history. So one of the things that I have suggested for the two other white paper groups that I'm working with is that a robust, big review journal article to be submitted to the journal Astrobiology be prepared previously to the white paper. So what this allows us to do as a group is to really dig in and fully flesh out our current group thinking about the scope of astrovirology, its impact on the field of astrobiology, what are the potentials for astrovirological observations or theorizing even as they apply to upcoming mission opportunities. And of course, I would not, even though this is in the planetary decadal, some of you may not be aware that there are plans afoot to move space biology into the Science Mission Directorate. Well, if you're not a bureaucrat, you probably think, who cares? But it makes a big difference in terms of how money flows and how things get managed. And so in terms of the impact on astrobiology and where it overlaps with space biology, there is a big overlap that has been artificially separated for many decades within the way NASA does business. And so clearly viruses, because we know they play such a major role in us as entities, us as astronauts, the environment, the microbial environment of spacecraft, and potentially human habitats on the Moon and Mars, there is significant crossover as it applies, I think, to the virus concerns that we have about Earth biology in the space context. So I just want to set the scene for that. I'm hoping that you guys will be enthusiastic about putting together a paper. I'll do everything I can to facilitate it. Obviously, since it's not my field, I have little specific intellectual content to donate, but I do have my understanding of the way NASA works and the way astrobiology works, and I think I can help with how we can make the biggest impact with this. And then my envisionment and the core group that I've been working with here, we're envisioning a distillation of the very high-level messages from that paper construction process, making it into the white paper. And we're on a very short timeline, as I said. It would be nice if we had essentially the paper already completed and submitted to Astrobiology, the journal, well in advance of February, so that we can at least cite ourselves in the white paper, if not anything more. And also, I've been in talks with Sherry Cady, who's the executive editor of Astrobiology. I have served forever on as a senior editor for Astrobiology. So she knows that this and other relevant white papers are coming and is very enthusiastic about finding a way to publish them in as timely a fashion as possible. So that is the bureaucratic and political framework and my personal interest. The other folks on the core team that have been trying to put this together, of course, Ken Stedman, who is on the screen, and Ken has been the fearless leader keeping astrovirology alive within the astrobiology realm through tireless and often unrewarded efforts over the years, and hopefully we can change that. And then my lovely young colleagues who are here in the room, Gary Trubel and Lawrence Livermore. Sorry, we have two Lawrences here. and he's a postdoc there and a wonderful virologist, and Kathy Bywaters, who has many talents in astrobiology and who is here at NASA Ames as a scientist. So with that, let me turn it over to Ken, who has prepared some initial remarks. Welcome, everybody, and I'm delighted to see you, and I plan to learn a whole lot today. Thank you.
Hey, thanks, Penny, and welcome to everybody. Particular thanks again to Gary. Huge thanks to him in terms of really getting all of this together. Thanks to everybody else, particularly the speakers. I'm not sure how many of you are online already, for agreeing to work with us on this. A lot of this was really kind of last minute. So again, hugely appreciated to all of those people who are going to be participating later on today. I think pretty much everybody knows that this is going to be recorded. A couple of things about Zoom and just generally how we're going to be organizing this. Some of you have already found the chat window, which I have just opened up here on the side. If anybody wants to go ahead and tweet this, hashtag Astrovirology. Let's see if we can get it trending, or maybe get it to go viral, as the case may be, over the next couple of days. That would be absolutely awesome. We shall see. I'm not going to make any guarantees about that, and I didn't pay them to support it either. So a couple of other things, given hopefully all of you have seen the schedule, we've scheduled a half hour for each speaker. Actually, we've really scheduled 20 minutes for each speaker, and then 10 minutes for questions. And there are a couple of reasons for that. One is we want to give people lots of chances to ask questions. Number two, speaking for myself, I know I have a nasty tendency to go over my time limit, so that gives us a little bit of flexibility, but only a little bit of flexibility. The other thing is, since we are trying to do this as multinationally as possible, that's partly why we're doing a half day this morning and a half day tomorrow afternoon, is to allow people in different time zones to be able to get into this. But part of that also means we really have to try and keep the time. And so if people actually do end up finishing early, shock horror, or the question period doesn't go quite as long, that gives everybody a physiology break for a couple of minutes. You can go and grab some coffee, you know, whatever it is. So we can start in each of the half-hour windows to make sure that everything starts on time and then hopefully ends on time. And the ending on time thing, Hopefully Marco will just, completely cut everybody off after half an hour and switch us over to whoever the next person is. I'll defer to him and say it's all the technical people's fault, rather than me being Mr. Nasty and coming in and saying, no, this is it. We've got to cut everything off. And so that's sort of, I feel, my role. In terms of the questions, anybody who's on Zoom should be able to ask questions directly there, but also you can type in a question into the chat, and then one or other of us will then actually read it to whoever the speaker is, and so they can answer those questions. And so those are the kinds of ways that we'd like to try and keep this thing running. Bear with us, particularly with me. This is my first workshop without walls, first sort of virtual meeting. So let us know if stuff is working or not working, etc. If it's a technical thing, maybe you could pop something into the chat. Hopefully somebody can help you with that. If it's a non-technical thing, things you like about this process, you don't like about it, please give us some feedback, and so that we can think about how we can do this better in future. And one of our presenters later on said that this is a really great way to lower our carbon footprint in terms of putting together meetings, and again, hopefully widen the audience, make this a lot more accessible to lots of different people. So any comments, thoughts, etc. along those lines, we'd really greatly appreciate. And as Penny mentioned, hopefully we'll be able to lean on as many of you as possible in terms of trying to put together a review article, putting together a white paper, so that we can really kind of advance the field of astrovirology beyond the four walls that you can see behind me right here, and a few of my colleagues as far as that's concerned. So before I have a little presentation, I just wanted to open things up for any sort of technical general questions before we sort of move into the formal parts of the presentation. So does anybody have anything that we want to try and clarify before we move into that? Otherwise, I've got a couple of slides on the virus focus group. So I get a quick question on the chat group. Can we share the workshop link via Twitter? I think absolutely. Please.
Yes. Yes, please.
Someone else on the chat room says it's too late. They've already done it. So thank you. Yeah, no, the idea is to make this as widespread as absolutely possible. That's sort of proselytizing as much as we possibly can. Okay, any other sort of question kinds of things along these lines? Okay, let me see if I can share my screen and give you a couple of slides on what we've thought about astrobiology in the past. So let me see which one we've got here. Okay, hopefully, can you see my screen here? Okay, good. So this is, speaking of articles in astrobiology, hopefully some of you have seen this. I suspect very few of you have actually seen the cover picture, because it seems that nobody looks at covers anymore. But this is the article that Aaron Berliner, Tomohiro Mochizuki, and I put together basically a couple of years ago, and it came out right at the beginning of 2018. And really sort of I'd like to think of it as sort of a backbone to think about astrovirology and how it could be fitting into a larger picture in terms of astrobiology as well. Right after this came out, many of you may have seen that, let me see if I can get this to move. Change slides. How do we get this to move slides forward? There we go. Most of the press actually thought this is what astrovirology is. So I think this is one thing that we do need to be careful about. Whenever I talk to the general public about astrobiology, the immediate thing is, oh, should we be scared? Now, this particular one, unfortunately, in the original, it's actually an animated GIF where you have the quote viruses, which actually look to me a lot more like macrophages, bouncing back and forth against this background. It's really frightening, I guess, is one way in lots of different ways of thinking about the term. So I'd like to think much more about this in terms of astrovirology, excuse me, rather than this in terms of the killer alien viruses. And thanks to Penny for sort of bringing up the origins of sort of thinking about viruses in an astrobiology context. This was really the brainchild of Barry Blumberg, the late Barry Blumberg, and he actually founded the virus focus group. I believe, remember correctly, it was in 2002, so about 17 years ago now. And the basic take home of the virus focus group was Barry wanted people to think about viruses because he was really excited about viruses, and so was I. And then when Barry actually asked me to be co-chair of the virus focus group as a beginning assistant professor, when a Nobel laureate asks you to be co-chair with him of something, you basically say immediately, yeah, what should I do? The other side of being co-chair with a Nobel laureate and someone who's running the NAI means that you're co-chair, it means you basically end up doing almost all of the work. So that was sort of the process. And really what we were mostly interested in was the origin and evolution of viruses. That's why I've bolded them on here. And how viruses may have influenced the origin and evolution of life, here, there, and everywhere. And so that's what we're going to be hearing about in the talks in the next, 2.5 days in terms of thinking about where viruses fit in the bigger picture of astrobiology. But this also gets to the point of, what is astrobiology? And you can sort of think about astrobiology, I think, also in the larger context, is astrobiology is really kind of biology as well, just with a little bit of an astro context that goes with it. So until now, the virus focus group has basically been putting together workshops, symposia, field trips, and as I say, unfortunately, as Penny mentioned, we didn't get to do the underground workshop field trip in astrovirology, which would have been, I think, absolutely fascinating. But we really concentrated on sort of the things that we knew a little bit about, and also trying to get people from different fields of virology to start thinking together about, the bigger picture again, astrovirology. And so this, again, completely ties into the idea of a white paper, ties into the idea of putting together a review paper to try and start to tie together some of these strings which have been really very independent in terms of different virologists thinking about these things. As Barry always used to say, he wasn't really a virologist. I always say I'm not really a virologist, but I think most virologists would say, well, I'm a this or a that or a whatever. So, actually what is astrovirology? And tying that together, I think, is a really important aspect about this whole point of a white paper and putting together a review article. So what we've mostly talked about before, and again, a lot of this is going to be coming back up today, evolution of viruses. The viruses are absolutely everywhere. Very few people, even a lot of scientists, even a lot of real virologists, don't have a good handle on how absolutely ubiquitous viruses are. The whole area of virus ecology is really taking off. We're going to hear from a couple of speakers about what's going on in terms of viruses' role in ecology. Virus diversity, particularly in terms of structure. I'll talk a little bit about this in my talk at the very end. And one of my favorite lines, again, particularly in terms of an outreach point of view, is that viruses have a bad rap. I think there are lots of viruses that do good things. So far, all of the work in virology has been thinking about pathogenic viruses and the roles that those have, vast majority. And then finally, the only sort of real direct connection to astro and space, and again, thanks to Penny for bringing this up, has been the idea of how could viruses, and very specifically actually the extracellular form of viruses or virions, survive in a space environment. And this brings all kinds of ideas about panspermia, but also space environment in a much broader picture, other extraterrestrial environments as well. And this, I think, is an area, and we can talk more about it in the next couple of half days, is really, I think, a very open area for research, and something that personally I think that the field should be doing a lot of more work on. So I've got a couple more just really quick, so fun things of what we have been able to do in the past. Again, a couple of workshops. First workshop we had was here in Portland, and hopefully you can see Barry, those of you who remember knew Barry right here in the middle. Again, very much the focus of this whole project. Some of you may recognize some of the other people here. Forrest Rower has been a real leader in virus ecology. When he first came up and asked me where to find the NASA meeting, I wasn't sure if he was supposed to be here or a lost guy at some rock concert. Those of you who know Forrest would appreciate that. And Paul Turner, Greg Stewart, a lot of the real leading people. And then right here hiding at the back, or not completely hiding, of course, is Sherry Cady. We had an AppSciCon meeting. This is one of my favorite astrovirology slides. Roger Hendricks came up with this, lining up all of the viruses end to end. Ends up being, the viruses on Earth ends up being something like, a few million light years, so a ridiculously large number of viruses. We also had a really fun trip and workshop to Mono Lake, where we were actually able to go out on Mono Lake itself, visit Pahoa Island, which has a number of very interesting hot springs. Very near there, Mammoth Lakes, the Long Valley Caldera also has some really interesting hot springs. So we were able to sample there. Again, Barry, even though he said he was getting seasick, seems to have really enjoyed this particular trip. And Mono Lake is really flat, very few waves. And then our last field trip was to Lassen Volcanic National Park, which is one of the main field sites that I work in, and also had the opportunity to go to the Hat Creek Observatory and with Jill Tarter and talk a little bit about SETI. So it's really been a whole combination of different aspects of thinking about viruses in a bigger sense. And the last thing I wanted to mention is that we've also been very involved in outreach and trying to reach a lot of other people. And I'm noticing that it is now 8:30. So in terms of keeping on time, let's see if we can switch over to Steve Benner, assuming that he is actually online here. And I'll actually let Steve introduce himself, because I keep forgetting which institute he's associated with. So thanks to everybody. So Steve, is Steve around?
Yes, yes, he is.
Good.
If you don't mind, I'll unmute your microphone there. So Steve, we should be able to hear you now.
Oh, I was unmuted by the host. Okay. Yeah, I am unable to drop my power presentation onto the screen that I'm able to share in Zoom. Which I'm not quite clear why that's the case, but and for some reason Zoom has blocked out on the control panel all my things. So can you actually, for me, change slides?
Sure, I will do that. Let me bring those up really quickly.
Okay. Excellent. I don't fit. My talk really does not fit into this session, as I looked at all of the other ways in which people were talking about things. This, I think, has nothing to do with viruses unless you wanted to detect them on on Earth. So now, am I going to say to you, please change slide, or
Yes.
will I go back one slide? All right, don't go back one slide. The first slide. Okay, so keep in mind that NASA is very much interested in, well, the public is interested in, and the public is NASA's customer. Obviously NASA provides to the educated elite, if you're not a highbrow, you watch World Wrestling Federation for your entertainment. NASA provides a sense of mission, a sense of entertainment, and a sense of purpose to the general public, those who are familiar with this kind of things. And those are people, particularly who know Carl Sagan is. The one question that is at the top of everybody's mind in the public when you do surveys is whether or not life is out there. Well, we don't know quite what life is, but there's this concept of a self-sustaining chemical system capable of Darwinian evolution. And that is a theory definition of life that Carl Sagan was very much instrumental in pushing. And it's a theory definition. I'm using the formulation of Carol Cleland here, who was the astrobiology resident philosopher, because it is a statement not only about sort of definitional terms, which are very hard to pin down, but it really is a statement that when we adopt this theory definition, we are saying what we think is the only way to generate matter, as organic matter or whatever, having the properties of value in life. And that only way to do it is by this Darwinian process of replication with errors, where the errors are themselves replicable. And then natural selection does the rest. So your goal right now is to go out and find systems capable of Darwinian evolution. Chris Chiba and various other people made the comment that this was not an operational definition, because what were you going to do? Fly to, Mars, land, and then sit around and wait for something to evolve? Well, that's not a correct way of looking at this, right? You don't have to wait for a rabbit to evolve when you have a rabbit in your possession to make, or a pair of rabbits in any case, to make the statement that they have the capability of evolution. And so what you're looking for is molecular structures that have that capability. You're not looking for the actual evolution itself. All right, so the next slide is going to say what Darwinism needs by way of informational polymer. And there are two criteria that are both extremely important. One of them is the so-called Schrödinger aperiodic crystal structure. And that means that the genetic polymer must be able to change its information content without changing its structure. That is, all the building blocks have got to fit into the same packed form. There's a second requirement, is that the genetic biopolymer must be able to keep its properties constant with changing information. And so the next slide shows you the picture of Erwin Schrödinger. He wrote this book in 1943. What's remarkable about this book is that he did not know the structure of DNA. It was not to be Watson-Crick double helix only came out 10 years later. But Schrödinger was a physicist. He was famous for the Schrödinger equation in quantum mechanics. By 1943, he was slumming by doing biology. That's sort of what many physicists did at that time. And what he knew was that simple binding cannot guarantee faithful information transfer, at least not at the level that's needed for biology. Because a simple binding gives you a titration curve, which has a classical sigmoidal shape, which if you ever did any work like this in your biochemistry laboratory, you're familiar with. And what you need to do to get faithful replication is sharp phase transitions between a correct pairing and an incorrect pairing. And for that, Schrödinger said we have to rely on what's called the physics of phase transitions. Now, I won't go into the physics of phase transitions, but the melting of a crystal is an example of this, which everybody's familiar with. So if you take ice at minus 0.0001 degrees centigrade and then move it to plus 0.001 degrees centigrade, the entire ice cube goes from a solid to a liquid in a phase transition. And by the way, impurities in that ice crystal will cause that melting point to be lower and broader. And so the physics of phase transition is what allows very precise exclusion of impurities from a crystal. It also is what you're relying on in DNA to exclude impurities, that is mismatches, when you're doing DNA replication. And for that, and this is the key point, the exchangeable informational building blocks must all have the same size and shape. They must all fit into, now, obviously a crystal of ice, all the water molecules are identical, and so there's really no information in the crystal structure of ice. But what Schrödinger said, okay, great, what we can do is make the molecules that are in that crystal a little different. Therefore, it's aperiodic in terms of its placement of these building blocks. The aperiodicity is what contains the information, the genetic information. But in order for this to work at all the building blocks have to fit into exactly the same structure. So you go to the next slide. This is, of course, actually, I wish I had control. I'm going to go to the next slide for me. So I should put in here somewhere a structure of DNA. That's a perfectly good structure of DNA on the left. Let's go to the next slide. The next slide. Yeah, there you go. So I've put here the GC, a CG base pair in the upper left-hand corner, and the TA base pair, the next one down. As you can see, these are essentially the same size and shape. I've actually fixed, for those of you who are going to take a test on this subject, I have actually fixed the structure of A to put it in an amino group down to form a third hydrogen bond with T. But the key point here is that GC and CG and AT and TA are all the same shape, and so if you replace CG by TA, you don't actually change dramatically the structures of those molecules, and these can fit into a aperiodic crystal structure. And it's interesting that when Watson and Crick came up with their structure, for those of you who are not familiar with the history, briefly, Linus Pauling was in competition with them. Jim Watson has written a book which very much describes how they were afraid that Linus Pauling, who was at the time a very famous scientist, was going to beat them into coming up with the structure of DNA. Linus Pauling actually did not beat them because he was putting phosphates of DNA inside the backbone when they were really on the inside the structure that he was assembling, and he was building a structure for DNA which had 3 strands of DNA, not 2 strands. And Watson writes in his book how any biologist would know that the important number was 2, not 3. But when Watson and Crick finally got the structures of the GC pair correct, which they were trying to build a model with an incorrect structure of G, the first thing that they said when they looked at those base pairs was, aha, this fits Schrodinger's aperiodic crystal structure. That is, we can exchange CG pairs for AT pairs, or CG pairs for TA pairs, or CG pairs for GC pairs, and not change the overall structure. So that's the first thing that they recognized about their model, was that it fits Schrodinger's criteria for aperiodic crystal structures. Now, the other structures on this pair are all synthetic biology structures. These are molecules that we have made, and you can see we are changing the hydrogen bond donor and acceptor patterns. We've written on these structures blue hydrogens as hydrogen bond donors. So C as a hydrogen bond donor acceptor pattern, blue, red, which pairs with G, which has a red, blue acceptor donor hydrogen bonding pattern. And T is a small thing with a red hydrogen bond acceptor donor acceptor pattern, which pairs with a big amino adenine, blue donor acceptor donor. And the rest of the structures here is there are 8 more nucleotides. None of them are actually natural as far as we know. They shuffle the hydrogen bond donor and acceptor pairs, but they keep the size of the pair uniform so that you can replace anything, any pair, any of the 6 pairs on that slide by any of the other 5 pairs, or the 6 pairs in the reverse direction, and not change the structure. And this artificially expanded genetic information system is, in fact, perfectly capable of meeting the Schrodinger aperiodic crystal structure. So the next slide, I think, maybe has, yeah, there you go. So these are crystal structures at the bottom. You'll see B, C, D, E. Well, C, D, and E are 3 different structures of 3 different DNA duplexes built from, in this particular case, 8 letters of genetic alphabet, but all still fitting the Schrodinger aperiodic crystal structure. And the structures are all the same, C, D, and E. And if that doesn't seem so for you, the structure B has all of them placed on top of each other. So let me just pause there and make sure everybody understands the Schrodinger aperiodic crystal structure. That is, to have a Darwinian molecule, you got to be able to replace the informational units without disrupting a crystal. How are we doing? Is anybody there?
We're good.
Okay, so that's the first requirement. Okay, next slide. Let me just see if I did this right. Yeah, and so there you go. So the first thing is that Darwinism needs is a genetic system able to change encoded information without changing its structure. That's Schrodinger. And by the way, homochirality which is sought by many of the life detection missions that NASA is contemplating, including, for example, the mission to Europa, is actually not a primary attribute of living systems. It is a derivative of the Schrödinger aperiodic crystal structure, and is absolutely required for the informational biopolymers. But biological systems actually mix D and L amino acids all the time. In fact, gramoside, and there's lots of antibiotics that do this. What you can't do is have that system be the evolvable system as in an information sense of the term. So if you went looking for homochirality on Europa and you managed to land yourself in a pile of gramicidin, you would not see that as would not see homochirality as a life detection criterion, and you would mistake gramicidin as being the product of abiological processes. But homochirality is what you're going to see in the informational biopolymer, and so your problem when you do life detection in space, or for that matter in Antarctica, you do need to know that you're looking at an informational biopolymer before you start looking for homochirality. So that actually brings us to the next system, which is the genetic system. Not the structures of the building blocks must not only be robust with respect to replacement, so as to not disrupt a aperiodic crystal structure, but changing encoded information also has to be possible without changing the physical chemical behavior of the informational polymer, such as its solubility, its molecular recognition, its reactivity. And one thing we know from Terran biochemistry is that these systems are actually hard to find. So in proteins, for example, and I've used sickle cell anemia here as an example, a single amino acid change in a protein of 500 amino acids can cause that protein to precipitate. And that's in fact what's forming sickle cells in biological systems. So, hey, you know, if you had your genetic polymer where you changed a G to an A or a T to a C to try to get more fitness, and then the DNA precipitated, it would be a really difficult to evolve system. So you've got to be able to keep the reactivity more or less constant. So the next slide, maybe. Let's just see what I got here. Yeah, so the polyelectrolyte theory of the gene says that what you need to have to give you that stable physical properties with respect to information change that has had this repeating charge in the backbone. And the reason for that is because that to say a molecule is charged is the most important thing you can say about a molecule, right? Not only does it describe its solubility in water, which of course is night and day versus charged versus not charged, it also dominates all other electronic properties of a molecule. So a molecule can have a charge, that is a monopole, and if it doesn't have a charge, then you can start saying, well, does it have a dipole? And if it has a dipole, that's the most important thing you can say about a molecule. If you're like benzene and you don't have a dipole or a charge, then you can go down and ask, well, does it have a quadrupole, which benzene actually has? And that becomes the most important feature of the molecule that dominates its behavior. How are we doing? So a charge is big. If you don't have a charge, you can worry about charge separation, that is a dipole. If you don't have a dipole, you can start worrying about higher order structures. But the point about a polyelectrolyte is it lets you change the bases without changing the behavior of the molecule. And so if you go to the next slide, I just show you one of the empirical tests of this from our laboratory. We tried to make DNA without repeating charges. And I'm not going to go into great detail, but there's DNA on your left, and there is a molecule that we made at great expense and effort on the right. You'll see I've added a proton to the phosphorus to make sulfur. That removes the backbone charges. And what you'll discover about this molecule is that when you get rid of the repeating charge, what now happens is this molecule has a repeating dipole, and instead of behaving like a respectable genetic biopolymer, it starts behaving like sickle cell hemoglobin, right? It folds, and actually the next slide shows you, just for fun, a folding, unfolding thermodynamic property, right? I've got an 8-letter oligonucleotide, A sulfone, U sulfone, G sulfone, G sulfone, U sulfone, C sulfone, A sulfone, U, and you're looking at here a plot of the ultraviolet spectrum as a function of temperature. and you're looking at the unfolding of this particular molecule, because these molecules fold like proteins. Well, why? Because unlike DNA, they do not have a repeating negative charge. And like proteins, they have a repeating dipole, and so they behave like proteins. We're making a single change in the sequence of bases in that molecule will change its physical properties, including its solubility, dramatically. And this, therefore, is not an adequate molecule for Darwinism. How are we doing?
Good.
Next slide. Yeah, and so, I mean, I just sort of schematically put this out here. So you're looking at the repeating dipole in peptides or in sulfones. It's like putting a bunch of magnets on a string, right? North poles and the south pole, if you let go of the ends of the string, the whole thing will collapse upon itself. But in contrast, if you have a polyelectrolyte, that is a molecule with a repeating charge, here a repeating negative charge, but this also works if the molecule has a repeating positive charge, that molecule doesn't fold, and therefore it can template. And the next thing I should mention is on the next slide, which is that this, if you get rid of that repeating backbone charge, you also lose Watson-Crick rules. So here's just a structure of one of those sulfone molecules, which I've shown. We have a UU pair. The next slide shows you a little bit more graphically what's going on here. You've got two kinds of repulsion, right? You have the repulsion at the top of that structure where the double-headed arrows are showing repulsion between backbone phosphates of a single strand. And that's, of course, what keeps the molecule unfolded. And then you'll notice that the Coulombic charge repulsion between strands, what that does is lets the molecules, or forces the molecules. The Coulomb's law predicts, of course, that the backbones repel. In fact, as an experimental piece of evidence to support that, DNA duplexes are more stable in high salt, where the backbone charges are shielded from each other by salt. But what this backbone repeating charge does is drives strand-strand interactions as far as possible from the backbone. And that's what gives Watson-Crick rules. Get rid of the repeating backbone charges, as now many synthetic biologists have done. Those rules go away. Okay, so go to the next slide for me, please. Yeah, so the Darwinian informational biopolymers must be charged because, among other, in water at least, because it keeps the biopolymer dissolved in water. These backbone Coulombic repulsions force strand-strand contact, so the Watson-Crick edges of the nucleobases. That's what gives you the rules that you memorize in middle school: A pairs with T and G pairs with C. And of course, polyanionic structure discourages folding, and that's what lets you do template. All right. How about the next slide? And by the way, synthesis, again, tests this. I did not mention this when I put up all of these slides of all these structures that fit the Schrodinger aperiodic crystal structure, but everything that we do in this molecule also has that repeating charged phosphate in the backbone. So this is a perfectly good evolvable genetic system with 12 letters instead of 4. I'm not saying necessarily that life on Europa or Enceladus or in the water ammonia ocean beneath Titan will have a 12-letter alphabet, but they could, because they meet the two criteria required for Darwinism. And by the way, these are sufficient criteria. We actually don't have to have any other features of that biopolymer. It has to be a repeating backbone charge that can be a positive charge, and the building blocks must be exchangeable without changing their overall size and packing. All right, how are we doing?
Great.
Okay, no one's I've either killed you all or. All right, so the next slide says, all right, great. So this, by the way, just for fun, I just put some stuff in here just to show you how good a 12-letter genetic system is, as long as it keeps the Schrodinger aperiodic crystal structure in tow, and it keeps the repeating bonding patterns. We just published this paper with an 8-letter genetic system where we measured hundreds of melting temperatures, the thermodynamic properties of the formation of AT, GC, SB, and ZP pairs. The SB and ZP pairs you've never seen in your middle school classes, but there they are structurally. And If you want to look at the predicted melting temperatures on the right side, on the x-axis, and the experimental temperatures on the y-axis, the correlation coefficient is R² is about 0.87. This is just as good as with natural DNA. And by the way, the deviants from this are not the funny bases, not the funny nucleotides. The outlier is AT, okay, which, as you know, is joined by only 2 hydrogen bonds, and that creates no ends of problems when you're trying to design primers and probes and do biotechnology. But the point is that this is an evolvable system which is just meeting 2 requirements: Schrodinger's requirement and the polyelectrolyte requirement, and everything else can be changed in this system, including the hydrogen bonding patterns, the number of bases, the side chains appended to the bases, the functionality of the bases, any of a number of things. So again, I'm not necessarily expecting to see this on Enceladus, but you can see that the rules are predictive in the sense that we can go into the laboratory and make our own Darwinian systems just by following these 2 rules. Okay, next slide. Yeah, and I just am beating a dead horse. Next slide. Okay, now we want to use these features to build a life detection device. And one of the things that's very important to know is that if I have a polyelectrolyte, if I have it in dilute solution, it's very easy to concentrate that from a dilute solution. Well, how? All I have to do is put down a detector, which is at the bottom of this slide, with a bunch of positive charges on it. And that, of course, will absorb a biopolymer from solution with a bunch of negative charges on it. And because of what we call polyvalency, the repeating negatively charged backbone will displace inorganic ions like bromide with a single negative charge, or sulfate with 2 negative charges, or perchlorate, which unfortunately has 1 negative charge, but never mind. So it's very easy if I take a dilute solution to concentrate a polymer that meets the second criterion for Darwinism from a dilute solution, even a salty dilute solution. And next slide, please. And so you can imagine a device, in fact, we wrote a paper about this 2 years ago, which you can read, where you could put 2 plates, okay, in, and the 2 plates, one of them has a bunch of negative charges on it, one of them has a bunch of positive charges on it. The first one is supposed to capture Martian genetic polymers if the repeating backbone charge happens to be positive. The positively charged plate captures Martian DNA if its repeating backbone unit charge is negative. And so it will concentrate this from dilute solution. And then the next point we should make is that you want to be able to then ask or query what is absorbed to see whether it is fitting Schrodinger's requirement. And it's kind of interesting because if I take DNA and its building blocks, each of the building blocks is chiral, and so they will rotate plane polarized light. But if I hook them all together, the chirality is bigger. You have a super chirality, and that's because each of the little building blocks with the same chirality, that is homochirality, induces a bigger chirality. In this particular case, for example, in your DNA, a right-handed, not a left-handed double helix. And that right-handed double helix has more rotation of the polarized light. It's more optically active, if you will. It's superchiral because it has amplified the microscopic chirality of each of its building blocks. And that's actually something that can be detected by, in this particular case, making this thing that's absorbing the molecules a waveguide and putting plane polarized light through it. So if there is, in fact, a homochiral polymer absorbed, that plane polarized light will be much more rotated than if it's not homochiral. Next slide. Yeah, and so, then there's only questions about sensitivity, which I won't go through here, but obviously you can just do a Biot surface plasma resonance absorption to detect the absorption of these things. The sensitivity is low, the long path length is required. And we don't actually know what the refractive index is of Martian DNA, so you have a problem managing that. So the next slide is, of course, you can look for the ultraviolet absorption of the side chains onto that waveguide. That's a more sensitive test, but again, we don't know that Martian DNA absorbs UV light. The next slide is what I just mentioned, this sort of macrochirality. We can look for rotation of plane polarized light, which will be greater from a polymer of homochiral building blocks than rotation of the monomers collectively. Next slide. And by the way, none of this precludes downstream analysis if you're building your light detection instrument. So, you know, we can obviously collect the allegedly Darwinian polyelectrolyte polymer and then displace it later to do MALDI mass spec or some information to get some information about the detailed structure of the polyelectrolyte. Next slide. Yeah, so I mean, this is a complete architecture. It's a wonderful concept. You go to the Martian polar ice cap, or for that matter, you can go to Antarctica. You dump ice in the top. You know, the Mars cap is nice because it surveys the entire accessible surface by dust storms. So you don't have to do caching or make correct guesses about locales to sample. You got to thaw the sample, but you got to keep in mind that all the other steps that I'm doing are using energy, but that can all be added to the budget for the thawing budget for the energy of thawing of the material. You got to disrupt cell compartments. Now remember, I'm going to put this into a cyclonic sharpless centrifuge, and if I've got dust in there and cells dust, the abrasive dust will disrupt cells pretty much for free. This sharpless centrifuge, that central tube is spinning, and so all the minerals go to the outside surface, and some of those minerals, maybe a lot of them, are chiral, and they would interfere with the optical activity measurement, so they're being set out to the side. You can actually withdraw them for if you want to analyze them. And then you pass the liquid through your polyelectric light capture things, and you can sample any of these things microscopically, or you can do mineralogical samples. I'm delighted to look at that cyclonic filtrated minerals and see whether there's borate there. You can see metabolites in the evaporated waste, and you can look at downstream analysis of the polyelectrolytes. There's really no possibility here for false positives because there's no other way of getting a polyelectrolyte building block assembly with a set of building blocks that are homochiral except by the influence of Darwinism, I think. Of course, false negatives are a matter of sensitivity of the device. All right, that's all I have right now. I think there's no slide to follow, right? Yeah, there you go. So let me stop and answer any questions you have.
Hey, Steve, unfortunately, we're running a little short on time, so maybe if people have a chance to ask you questions, maybe they can do that offline or maybe in a chat window a little bit later on, if that would work for you.
No problem.
Super, thank you. Okay, we got Arvind online.
All right, can everyone see the screen and everything else?
Yep, hi Arvind.
Hi there, hi. Thanks Gary, Kathy, Ken and Penny for organizing this virtual conference. It's awesome to listen to a variety of different things already and some of the concepts put forward. So today I'm kind of going to be talking about some weird stuff that we've been noticing with single-stranded DNA viruses, and I'm going to take you into the world of single-stranded DNA viruses in quite a complex system. So, see. Cool. All right, so before I go into the whole field of single-stranded DNA viruses, I want to actually start tackling a couple of things which I think are very important, and these are concepts of evolution. And we all know that viruses evolve by three main mechanisms. And one is basically nucleotide substitutions, where you basically get mutations occurring, or some of which are deleterious, some of which are beneficial, others are pretty much neutral. And then to repair mutations that are deleterious by, again, mutating those sites is actually very complex, and to fix them is a very, very slow process. However, there are viruses that are very good at fixing a variety of mutations by a mechanism called recombination. And this usually happens when you have two viral templates within a cell. And what happens is there is template swapping that takes place during replication. And in this mechanism, you can generate chimeric viruses, chimeric genomes. And as a consequence of that, to repair a deleterious mutation is actually very, very beneficial. It's very efficient in this kind of a mechanism. But at the same time, if a virus is trying to explore sequence landscapes, then it is able to do this very efficiently using these kind of mechanisms. And then we have something that is very, very unusual but unique in the sense that we have reassortment, which takes place, which we commonly know in the field of influenza, where you have 8 segments that they can reassort over time. And as a result of that, you get different variations of at least the hemagglutinin and neuraminidase. And as a consequence, we have immunizations every year to try and combat the different variants that come through reassortment. But what is less known is that there are viruses which are segmented and multipartite viruses as well, where each segment or each genomic element that constitute the entire genome is packaged into an individual capsid. And there as well, you get reassortment taking place. So it's almost like each chromosome is packaged into an individual virion, and those all have to act together within a system to actually create an active infection. So if we kind of unpack all of this and start looking at viruses in a general context, viruses are very unusual in the sense that the genomic material can vary from DNA to RNA, and also in terms of polarity, and also whether they're double-stranded or not. There also is topological differences, whether they can be circular or linear. Things that is really unique about single-stranded DNA viruses is the vast majority of them are circular viruses. However, there is something that is very amazing, is that they actually are able to infect all domains, and you're able to infect archaea, bacteria. We know viruses that infect diatoms, fungi, a variety of invertebrates, plants, and obviously through metagenomic approaches, we're discovering viruses that we have no idea what they infect. But within these single-stranded DNA viruses, there are different conformations of genomic systems, where you could have monopartite or multipartite molecules. So basically, you could have one single chromosome into which all the genetic material is packaged, or you have multiple chromosomes that then carry different genetic information, and they're all unique in that context. But one thing that we've really focused on these viruses primarily is because we're keen on understanding evolution of these viruses, and these viral genomes are relatively small, so we can actually sequence them to completion, and we can look at coding and non-coding regions to figure out what the variations are. Now, one of the unique things about some of these viruses, single-stranded DNA viruses, not all, but a vast number of them, is that they have certain very, very unique properties. And one of them is an enzyme called replication initiator protein, which actually has 2 domains. It's got an endonuclease domain and a helicase domain, which initiate the whole mechanism of replication of these circular molecules. And very, very unique motifs, which are very recognizable across the system. But if we were to break those 2 motifs and draw some phylogeny, infer phylogeny based on that, based on all the single-stranded DNA viruses that encode these kind of proteins, we notice that the phylogeny is not congruent. And that kind of suggests to us that these viruses have at some point exchanged material or are recombinant even at a protein level. So some of this, obviously, we can't detect at a nucleotide level. We can see it at a protein level because of these variations. But the other thing that is staggering about these viruses, and these are nucleotide substitution rates that we've calculated, other people have calculated. We've calculated based on lab experiments. We've made infectious clones, put them into a system, measured the mutation rates over 15 years in some cases to see what's been going on. And if you look at them, that all across the theme of single-stranded DNA viruses, there appears to be a theme that the mutation rates are really high, or the substitution rates are really high. And in some cases, they're as high as those of RNA viruses. And this general hypothesis has always been that RNA viruses are actually error-prone because they use low-fidelity enzymes. Whereas here, you've got DNA viruses which are using host DNA polymerases, yet they have such high mutation rates or substitution rates. So that's kind of something that's really been bugging a whole group of us. So now I'll start delving into small groups of viruses and take you through a journey of what we've been discovering in the single-stranded DNA virus field. And so there are these viruses called Gemini viruses that infect plants, and they're unique in the sense that if you look at the topology of the genomes, even though they're single-stranded, they bidirectionally transcribe. So that means the complementary sense is required to transcribe some of the open reading frames. But at the most, they encode about 5 to 6 proteins. But within this group, at the top left-hand side, you can see there are some things called begomoviruses, and they can assume 2 conformations in the sense they can be monopartite or bipartite, where they have 2 different molecules, which are roughly the same size, about 2.7 kb, get packaged into individual virions, and they have to work together. But when we start unpacking these things and we look at the 2 main prominent proteins that they encode, which is the replication-initiated protein and the capsid protein, and we infer their phylogenies, we notice that in a vast number of cases, those phylogenies are not congruent. And that straight away tells us that these viruses are very recombinant in nature. They recombine very commonly and evolve through mechanisms of recombination. So at one point we thought, okay, let's study mechanisms of recombination in these viruses. So if you start looking at viruses in general and you want to study recombination, there are basic hypothetical things that you can put forward. You take 2 genomes that are, say, 10% diverse from each other at a nucleotide level, chop them in half, make reciprocal chimeras, and put them into a system, into a host. And your outcomes can be a simple recombinant, which is a 2 breakpoint recombinant where a small piece inserts, or you can have complex recombinants where you have lots and lots of recombination events, so you get a mosaic pattern across the genome. On the other side, you can also have unique subgenomic elements where you get deletion and insertion of unknown sequences. In most cases, could be even host nucleic acid that gets integrated. And when you analyze all of these genomes, there are going to be fitness trade-offs. And you can measure fitness trade-offs, which genomes are beneficial, which ones are not. So we set an experiment together where we thought, okay, let's take 2 viruses. These are 2 grass-infecting viruses, and maize is a grass. And we found these in Africa. There were a problem within the maize industry, so we were working with this. And we took 2 viruses that were 10% different from each other at the nucleotide level. We chopped them in half, gave the other half to the other individual, made reciprocal chimeras. And then we put them back into maize genotype. And the viruses were able to recombine and find an optimal solution, which is found in nature today within 30 days. So they were able to explore 10% sequence diversity in 30 days and get to where they needed to be and what is present in nature today as an optimal solution in 10 days. And if we change the variety of maize, so basically you've got some sort of genetic traits that we embed into this for selective breeding, what you notice is that the maize plants are very, very sensitive to these kind of things. So they have low tolerance to the virus. They're very susceptible. The viruses seem to be exploring a bit of sequence space, but still aggregating to an area which is very, very common down here, which is the main solution. But if you put it into a resistant variety, pretty much everything goes to that one solution that is required in nature. So this is very staggering that in nature, this evolution experiment has taken place over 250 years. We've done it in 30 days in a lab experiment. But also we noticed there is some level of genome plasticity within these molecules, these viruses. So here is an example of a Gemini virus that's bipartite, and you will notice that we've been discovering these groups of viruses where this entire segment has been incorporated into this molecule to generate a larger molecule that still infects plants. So one thing that we can't figure out is whether this is an acquisition or these molecules have been derived from a deletion of this segment, and that's how these viruses are evolving. So there is a variety of different things. And so within this context, how do you start pairing these molecules together? Like, is this and this totally related? We can do that by there are these common regions, we can look at them, and because one is required to replicate the other, we're able to do this. But it's also very complicated, so we're trying to come up with new methods to understand how we can put these pairings together, and some of them are based on network analysis But when we even start looking at phylogenies of these paired molecules, there isn't any congruence. They're pretty much exchanging or trading with each other very openly. So that leads me to another group of viruses, which I think are very, very important, because these are the multi-component viruses, again infecting plants, but we're discovering similar viruses in eukaryotic systems. And these molecules are 6 to 8 molecules. Each one gets packaged into an individual virion, and each one encodes one single protein. So in this case here at the top, there is a protein that initiates replication, replicates all these molecules. Here is a molecule that encodes a capsid protein that needs to encapsulate all of these. So you can imagine the sheer dynamics of these molecules having to interact with each other and inside a cell system. Also, the delivery is also important because at some point within a host, you need to get all of these into a system so they can all cooperate and produce progeny. So we started studying them in greater detail, and we started noticing that they were reassorting, very much like influenza viruses. They're able to trade molecules between each other very openly. And what was really staggering is that when we look at individual molecules between themselves, they were recombining very rapidly. And we take these molecules that are all different, and we analyze them for recombination, and they're also recombining in these common regions. So they're actually exploring sequence space at a completely different league, which is really fascinating in the sense that these viruses are just like viruses that are on Red Bull and acid, and they're just out there to explore sequence space, and they're going to do it no matter what. So the other thing that we were always fascinated by is why are these viruses single-stranded? Because single-stranded nucleic acid is not stable. Double-stranded nucleic acid is stable. So we assumed that they would be forming complex secondary structures within the genomic elements when they're packaged into genomes. So we analyzed all the sequences of single-stranded DNA viruses that were available in public databases, did folding analysis, so basically in silico analysis of folding, and tried to look for paired sites that we saw very commonly. And when we start looking at these paired sites in our in silico folding experiments, what we noticed is that purifying selection is strongest at paired sites, which is not surprising. We also noticed synonymous substitution rates are low at paired genomic sites, not surprising again. In our evolution experiments, we noticed that the mutations tend to accumulate in unpaired sites. Makes sense now. But what was staggering is that we saw evidence of complementarity to evolve base pairs. So there is some sort of complementarity involved in it, that if one nucleotide here in a base paired region it mutates, there is a compensatory mutation that takes place to retain that particular structure. So we started noticing that these single-stranded genomes fold into complex secondary structures. Those complex secondary structures are very unique, that there is some sort of coevolution taking place of paired sites. And so this had never been shown, and this was kind of really interesting. And this also suggests that the secondary structure is highly likely very important for either regulatory mechanisms or maintaining some sort of genomic integrity within capsid proteins. And now I'm going to take you away from the plant side of things and go into an area which I think is interesting because it involves human activities. And here is a virus that I've worked extensively with. It's a virus that infects parrots. And what it does in the parrots, it's again a single-stranded DNA virus. It encodes only 2 proteins. It's about 2 kilobase in size. But it actually induces physiological changes in an animal. It suppresses its immune system. The animal's response in a lot of ways is feather loss, anemia. In severe cases like this one here, there's a sulfur-crested cockatoo that has pretty much lost most of its feathers as well as its beak. And, you have to give it to the Australians for writing beautiful obituaries about the birds. And this is Cocky Bannet that traveled around the world, died at the age of roughly 120 years. So parrots do live that long, and spent the last 20 years in a pub, and that's what it looked like. And so the phylogenetic tree on the right-hand side kind of suggests some very, very unique things. That here, what you have is some sort of a pet trade system taking place. There are breeding facilities from which there are pushing some of the legal and illegal trafficking of animals to different zones. And then what you're getting is establishment of new viruses and new ecosystems that haven't been before. And we've analyzed some of the facilities in Poland, for example. We worked with veterinarians there to look at recombination rates in some of these facilities, and a variety of different parrots. They come in from different parts of the world. They're brought into these aviaries. The animals are stressed. And before you know it, the virus is just circulating and being exchanged. And here we've got a genome of a virus that is more than 2/3 a recombinant that we identify in these kind of systems. So these legal and illegal trade systems are also facilitating the spread of these viruses and evolution of these viruses at a much rapid rate than people have thought before. And now we go into metagenomics kind of systems, and we start realizing that actually we know very little about single-stranded DNA viruses, because through metagenomic approaches, we just keep finding viruses, a lot of which we don't even know the hosts for. And here's a big group of viruses that we've recently established as a family-level structure. However, these viruses, the first group of them were found to infect fungi, induce hypervirulence, but we find them in pretty much all different systems. And you can see all the different kind of sample types that have been identified. And they've got a huge diversity. And so these things are really abundant in nature. But what is staggering again about them is that they encode two proteins, and we try to draw the phylogenies of the capsid protein and the replication initiator proteins, and not congruent at all. These are highly recombinant viruses that are present. But we find related viruses in nature, and here is something we found in an I-score. This is kind of work from Terry Young and Eric Calvert's group that I was involved with. And at about roughly just short of 200 meters depth at an I-score, they identified the virus, and we kind of worked together to try and figure out what the virus was. And then we looked at it and thought, wow, this looks very, very similar to a plant-infecting virus in terms of its genomic elements. So with Bob Gilbertson at UC Davis, we generated an infectious clone, put it into plants, and lo and behold, this virus was able to replicate and spread. But we dated this layer, and it was dated at about 700 years old. So we resurrected a virus that was 700 years old through reverse genetics. So this also suggests, and should be a caution for us, that the climate change impacts are real in the sense that we're melting permafrost at a very, very rapid rate. And so there are pathogens and organisms that are trapped in these ecosystems that are now being released into environments. And some of these environments haven't dealt with these organisms. And so what are the consequences going to be in future for either displacing certain organisms out of these ecosystems as these permafrost layers thaw? So if we take back a bigger picture and go back a few steps and go, all right, so we know all of these viruses, single-stranded viruses that encode the replication initiator protein, have these two motifs. But some of these motifs we also identify in fungal elements, in plasmids. And so we can start building network-like analysis. And if you just take the helicase domain, and we know helicase domains are present in a lot of different systems, including replication machinery and repair machinery, we can start building networks of similarity. And we start noticing that some of these helicase domains are also found in other groups of viruses, which are circular double-stranded DNA viruses, but a variety of different plasmids as well. And from there, we can start then inferring concepts of where these viruses have evolved from. And you start noticing that it's not just one system. There is multiple acquisition and multiple events that have led to these viruses evolving from each other. And if you look at it at a much more broader perspective, we start noticing that some of these viruses haven't just evolved in a one clear lineage process. There have been multiple evolution processes from plasmids and plasmid-like ancestral sequences that have led to the emergence or evolution of these viruses in nature. And then what you can start doing is, this is something that I've been fascinated with, is that viruses all have capsid proteins. And the capsid proteins are very important because they recognize certain particular receptors to get into cells. Now, Ken and a variety of other people have found that there are certain viruses, single-stranded DNA viruses, that encode capsid proteins that are much more closely related to viruses, which are RNA viruses that have similar capsid proteins. So here is an example, is Tombus viruses are RNA viruses. Their capsid proteins are very similar to some of the capsid proteins that are encoded by single-stranded DNA viruses. So where has this chimeric acquisition taken place? At a cDNA level at some point. So these viruses have some sort of a proxy in terms of capsid fold space. And I think capsid fold space and Mart Kuprov is a big proponent of this, is that capsid fold space is limited. And as a consequence of it, is that there's only so much fold space you can have at a protein capsid level. And some of these viruses are then acquiring these capsid-like molecules from different areas. And so here are all the Gemini virus capsid proteins that we know about. And what is interesting about these is they actually have a capsid fold, which is very similar to that of an RNA virus, which is satellite tobacco necrotic virus. And you can see the vast number of unclassified circular DNA viruses that have this fold as well when you start looking at network analysis. So these network analyses are becoming very, very important in the way we understand. And so yes, complemented with phylogenetic trees, these are very, very good ways of pulling down related sequences and understanding more broader concepts of viral evolution. And so I want to kind of start ending this with a bigger concept. And this is that if you look at the phylogenetic tree on the right-hand side, you notice these are all single-stranded DNA viruses that encode replication-associated or initiator proteins. And the ones that are well classified are in these little triangles, collapsed branches labeled here. Everything else that's on this illustration here are taxa that we've identified in our research group. Now, everything that is in red is what we have identified in the Antarctic ecosystems. So to me, if I were to draw a plot of viruses that exist on this planet in terms of what we know about them today, they would be scattered on this plot on a 10 by 10 grid in various different places. And if I was to sum that up in terms of where our current knowledge is, it would be less than 1% of all the viruses we know on the planet. So that means we have a lot of work to do in terms of if we really want to understand virology. We're far from it. We've only had a huge bias to viruses that cause disease and viruses that cause disease in organisms that we dearly care about, like plants, animals, humans. Everything else we've just neglected. And also, as Ken mentioned, we've completely looked at it from a disease angle, never from a beneficial angle. And we do know that viruses are the most abundant entity on the planet. If they're so abundant, they should be having beneficial roles in ecosystems. And we do know that in marine ecosystems, they are very, very important for carbon sink. And at the bottom here is a very, very unique paper, which I think is great. And this is part of the efforts of the Tara Oceans Project. And if you look at in the epipelagic samples, that the water columns that they've looked at, they've saturated the amount of viruses they can find. So they've got to a level that doesn't matter how much at that layer they sample, they have got a very good handle of sequence space in the regions they've sampled. But when you go into different areas, they're still not close to it. And I think with single-stranded DNA viruses, I think we're in this stage that we're not even at all close to having figured out everything. So with that, I'd like to thank a whole lot of people who have been involved in a lot of the work that I do, that I've spent for the last 15, 20 years. And there are groups of people all around the world, as well as a whole lot of crew that have been students in my lab and are still staff in my research group. Thank you, everyone.
Questions, anyone?
Hey, Arvin, Siobhan here.
Hi, Siobhan.
So I posted this to the chat, but what's the real risk of these viruses being in infectious capsids as the permafrost thaws? I would worry about things like the Tabamo viruses, because those things can withstand incineration and all kinds of environmental challenges. But should we actually worry about intact virions that could actually be infectious from permafrost? Or are we just going to keep learning about what was there hundreds of years ago, and we could resurrect these things with reverse genetics, and that's very, very cool, but they're not actually going to infect our plants tomorrow?
Well, they could infect. So look at our pollution levels on the planet. Microplastics are pretty much everywhere. A lot of these viruses will bind to microplastics. Microplastics, they seem to get into systems. We've got a lot of detergents in our ecosystems. And so detergents mean you've got actually a beautiful surfactant that can deliver things straight into a variety of different organisms, especially single-cell organisms. And we've got vectors, insects that are pretty much picking up a lot of this material through their foraging behavior, and they will be taking in and injecting in. So the plausibility of a scenario like this is there. The frequency or how viable they are, I cannot kind of speak on that, but it is a plausible scenario.
Arvind, Ken here.
Yes, hi Ken.
Just a quick question in terms of the astrovirology, and also put this in the chat, and maybe Mart's the right person to ask this question of, but do you think that if there are limited numbers of folds that you can use for capsids, would that potentially be a way that one could think about virion detection techniques that maybe you could put on a spacecraft or something like that?
Yes, but it would need a lot of clever. Don't forget that protein sequence space is great, all right? Sorry, nucleotide sequence space is very great. Then you get into protein sequence space and you get into fold space. So you can have the same fold, yet you could have 30% diversity at a nucleotide level. So I think our detection in this kind of system is, unless we have ab initio approaches for fold protein, and ab initio approaches for detecting fold are very poor even today. So I think one thing that we really need to do is accelerate our crystal structure solving programs all around the world to solve structures of proteins. And once we have better databases, we can start spreading through homology modeling a variety of these protein structures. So that is one way, but there is a likelihood we might be able to come up with ways of using AI-based techniques to detect certain folds that we haven't really thought about. Anyone else?
So I think we're going to have to shift over here quickly to Britt. So let's thank Arvind for whirlwind of single-stranded DNA viruses.
Well, thank you.
Okay, can everybody hear me?
Yes, we can hear you.
Great, all right. And once I get this started, let me know if you can see the screen. Is the PowerPoint up for everyone else?
It is, we can see it.
All right, terrific. I must admit this is my first virtual workshop like this. I like it a lot, and I imagine that there's going to be many, many more of these in the future as we start decreasing our carbon footprint. So thanks for getting this going. So that was, I'm very pleased actually to be talking just after Arvind, because I don't actually spend much time on viral evolution, but it's hopefully now you've been suitably convinced that it happens rapidly and is important, and over longer timescales. And so what instead I'm going to talk about today is coevolution and why interactions between viruses and in particular bacteriophages are important in shaping bacterial populations and communities. And so just to bring everybody up to the same page, when we're talking about coevolution, of course, the first question is coevolution between what type of phage? And there are, generally speaking, three types of phages: lytic phage, temperate phage, or filamentous phage here in C. I'm going to ignore the filamentous phage, although I think they're incredibly neat, and focus primarily on lytic phage for this talk, but I don't want to ignore all of the incredible diversity that temperate phage introduce. And so I'm just going to spend a second here before shifting to my own work. And so if we're thinking about the impact that bacteriophages have on bacterial communities and populations, one of the obvious ones is through direct horizontal gene transfer and the genes that they introduce as they themselves integrate into a bacterial genome. And so these temperate phages, when they form lysogens in bacteria, can completely change the bacterial phenotype. And one of the ways that we care about this is if that happens to change bacterial pathogenicity, for example. But this is just an example. And of course, there are many different axes across which phenotype can change, potentially including colonization of spaceships, for example. But here, this is a really nice study, not from my group, but they looked specifically at this emerging opportunistic pathogen. They compared 795 genomes of this pathogen and found over 4,000 prophages. Let me just move this here. And so of those prophages, and I'm just showing this beautiful phylogeny they created, they did it with the proteome as well. This diversity is important in part because, as I already said, this is generating diversity in the bacterial population and community as well. They found that 78% of the genomes had, of the intact prophages, so these are prophages that remain capable of excising again and infecting a new host. 78% of them encoded potential virulence genes. And so if you want to understand pathogenicity and bacterial virulence, this sort of argues that ignoring prophages, which we quite often do, especially in metagenomes, could be a big mistake. Oop, let's see here. There we go. So if we go back here for a moment, as I already said, most of what I'm going to be discussing today is lytic phages here in A. And these are phages that are obligate killers of their host. So they inject their own DNA or RNA, hijack the machinery, must burst the cell in order to reproduce and infect new hosts. And of course, this has obvious ecological ramifications. It's killing bacteria. This was why people got excited about lytic phage when they were discovered in the 20s, because this was before antibiotics, a way to kill bacteria. But they do far more than that. And so if we want to understand bacteriophage, here we go. If we want to understand bacteriophage coevolution, one of the first parts of this is to understand how bacteria evolve resistance. So these phages are there, they're obligate killers, as I already said, so they're putting a strong selection pressure on their bacterial hosts. And this is just three very simple, the most simple examples and potentially the most common, but bacterial resistance mechanisms are constantly being discovered. And so here in A, you have a simple change in the receptor. So phages must bind, recognize a bacterial cell. And so, of course, the bacteria can change that receptor and instantly be resistant to that phage. It can no longer detect the bacterial cell. Or it could use these CRISPR-Cas mechanisms. So this is a mechanism by which bacteria can recognize specific sections of the phage genome and block translation of that, transcription of that phage. And then the last one here in C is apoptosis or cell suicide. The idea that once a bacterial cell detects phage infection, it actually kills itself before the phage can do that, protecting neighboring cells. And likely in any given microbiome or bacterial community, all of these mechanisms are happening all the time. Of course, you wouldn't have coevolution without the flip side. And you already heard a nice talk about viral evolution. In the case of phages, here's just one very nice example of how phages can counter-adapt against CRISPR-Cas resistance, CRISPR resistance. And so here, what you're looking at is at low density, this CRISPR resistance is actually quite effective. And so this is not work from my lab. This is work that was done in 2 separate labs. They find the same result. But actually, if you get enough phages infecting the same cell, the phages can essentially work together to overcome CRISPR resistance. And so this is one example of how phages counter-adapt. Of course, if it's a receptor modification, a counter modification would do the trick, a change in the tail fiber, and so forth. And so what I tend to think about when I think about bacteria and phage interacting is the constant arms race as bacteria evolve resistance and phage counter-adapt. And this, in theory, can go on indefinitely. Now, in practice, it's not clear. Phage genomes are small, and so there's probably are cycles, and there's going to be some sort of finite amount of evolution that can happen before you're almost back where you started, potentially. And so just to give you a few examples of why we should care about coevolution, this is beautiful work from Steve Patterson showing that coevolution, as opposed to just evolution, really speeds up the molecular evolution. And so this is a sort of in vitro test tube experiment here, where they either allowed bacteria and phage to co-evolve, so these are the C1 through 6, or just evolve. Okay, in this case, they keep one player fixed and they allow the other to evolve. And what you can see here is that this is just a phylogeny. You can see that there's far less sort of slower molecular evolution when evolution happens, when you fix one of the players and don't allow them to counter-adapt, than if you allow for coevolution. And the other thing they were able to show here is that this leads to rapid diversification, so rapid specificity and diversification. So here along the top in B, you're looking at the different coevolution lines and who those phages can infect. And you can see that, for example, C4 is capable of infecting a different set of hosts than C6. And so this happened within a very short period of time in the lab, and really, to me, emphasizes how important coevolution is, both at the molecular level and then at the phenotypic level. And this is work from Angus Buckling now, older work, where they just simply looked at bacterial phenotype, these different morphs, and they could again show that if you look in this sort of test tube experiment, if you allow phage coevolution in a test tube versus if you allow the bacteria to evolve alone, when the phages are present, the diversity, the allopatric diversity, so how different two test tubes, two populations end up being from one another, is far higher when phages are present than when they're absent. So rapid molecular evolution, rapid phenotypic diversification. Hopefully I've convinced you by this moment that coevolution matters if you want to be understanding bacteria. And one of the ways that you can measure coevolution, and I would say the gold standard way if you can do it, is using a time shift experiment. So I'm just going to spend a moment here because I'll show you some data using this, but this is just a theoretical model from a paper that Dieter Ebert put out a few years back, which is a great primer for anyone interested in time shift experiments. The idea here is simply you would co-evolve your populations and then freeze through time, or if you happen to have a permafrost sample, you can thaw through time, and do a cross inoculation where you're taking bacteria, let's say from one time point, and you're challenging that bacteria with phages from the past or phages from the future. And you have a clear prediction that they should be more resistant to phages from the past and less resistant to phages from the future, as those phages have counter-adapted to any resistance mechanisms. And this sort of directionality is indicative of these arms race dynamics in a co-evolving system. And you can do this in test tube populations, and this is a really nice one from Pauline Scanlon, where you, again, take your bacteria, in this case Pseudomonas fluorescens, and a phage, allow them to co-evolve over time, and do a huge matrix cross where you take bacteria here from different time points and you cross them with phages from different time points. And what this allows you to do is actually measure, and you can show that over time, bacteria increase their resistance to phage through those mechanisms I already described, but that phages are continually counter-adapting to that. And in this particular case, this is a true arms race because the bacteria are never, in the course of this experiment, once again susceptible to phages from the start of the experiment. Resistance continues to increase and phage infectivity continues to increase. So they're chasing one another through space. And again, we know this happens very rapidly in a test tube, but what my lab specifically focuses on is whether this happens in the real world and whether that matters. And we do that in two different ways. We do that in terms of looking at pairwise interactions between phages and bacteria. We focus on the plant pathogen Pseudomonas syringae. We have some new work as well on Erwinia and Lavora, the causal agent of fire blight. And then also just the role that phages play in the broader microbiome, in these microbial communities. And the system we use is the Phylosphere, above ground tissue of plants. This is an incredibly convenient system because the spatial scale is very clear. And so I'm going to start by showing you some work on the sort of more black boxy community level and follow and finish with some population specific data. The first experiment I ever did when I started to try to get a hold of what was happening in a natural system, the phylosphere of a horse chestnut tree in this case, is I went out and I just simply asked what bacteriophages are in these leaves and who are they infecting. And this is only focused on culturable components of the microbial community. But nonetheless, if you do that, if you culture bacteria from a given leaf, and then you filter that leaf juice and see whatever viruses are in there and ask who can they infect, you find that phages are far more infective. So this is a sympatric phage from the same tree. They're far more infective to bacteria from that same tree than they are to bacteria from a neighboring tree. And this just is a nice starting place. It's an old paper now, but it's a nice starting place because it tells us that in a natural system like the tree phylosphere, that phages are there, they're in high abundance, they're infecting about 15% of the bacteria in their local community, and that they're well adapted. And as soon as you have that evidence for adaptation, that suggests that they should be putting strong selection pressure on their bacterial hosts for resistance. So how do we test that? Well, I already told you the gold standard way to test it is with the time shift experiment. The downside of a black box approach like this is you can't follow a single lineage through time. So we're sort of glossing over, we're making a broader statement about evolution here as a function of turnover between species, turnovers between strain, as well as mutation. But nonetheless, we can take a similar approach. And we can ask, again, we take our leaves from these trees and we put them into the freezer. You can then resurrect the bacteria and the phage and say, for a given bacterial host, let's say here's just an illustrative example, a bacterium from July from tree number 2. And you can then challenge that bacteria with potential phage, so this filtered leaf juice, from earlier time points, for example, June here, the contemporary time point, July, or later time points, August and September, for example. So if we do that and we lump across the 8 trees that I used in this experiment, what we find is a similar result to what Pauline Scanlon and others often find in test tubes, which is to say a directional pattern where, in this case, if we look at bacterial resistance, oh, sorry about the y-axis there, something funny has gone on, but the bacterial resistance on the y-axis, we can say that bacteria are more resistant to phages that they've experienced previously in the season. So this is the past phage. Okay, so if they've been under selection from that phage, they are more likely to be resistant to it. The contemporary time point somewhere in the middle, and then future phages tend to have counter-adapted and overcome that resistance. And so another way of saying that is bacteria are least resistant to phages that they have not yet encountered, phages that have likely counter-adapted to that contemporary resistance. I should say that the solid bar here is just the average across the trees, and there is a lot of variation. Not every tree is behaving in the same way, but the pattern is highly statistically significant, even if you pull out this sort of super co-evolving population here on the far bottom. Okay, so far I've told you that in a natural system, phages are present, they're highly prevalent, they're obligate killers, so they should be putting selection on their hosts, and indeed it looks like bacteria are becoming more resistant through the season as they're encountering these phages in a natural system. What does that mean for diversity? So I've again given you some nice examples from a test tube that we have clear theory. The kill the winner theory is a good example of that, where we expect that phages should, as they adapt to common bacterial types, they actually take the fitness of common types down, and they should be increasing diversity of the microbial population or community. But can we actually show that to be true? It's a bit of a challenge. And again, as a first pass, we took a black box approach. And by we here, this work was led by Norma Morella, who's a PhD student in the lab who just recently finished. And so what she did was went out to the field, sampled a whole microbiome. So in this case, we used tomato. It's a very convenient model system. She was able to go out to fields, sample the whole tomato plant, wash off all of the bacteria and viruses from the surface of leaves, and then through a series of filtration steps, separate that microbiome into 2 components, the microbial component, the bacteria, and then the viral component. And the nice thing about that is then we can either inoculate sterile plants with either the bacterial community alone or that bacterial community where the phages have been recombined. And again, a very black box approach, but it allows us to get a really nice average of whether phages are generally making a difference during initial microbiome colonization in this case. And we use digital droplet PCR as a way of quantifying absolute abundance of our bacteria using 16S probe, and also for future studies that I'll show you specific bacteria as well. Okay, so she sprayed plants in replicate with either the bacterial component alone or the bacteria with its sympatric, the same phage from that community. And the first thing to say is when we look at the plants a day later, so a day after they're inoculated with this diverse microbial community, we can show that when you recombine with the phage, this orange bar here, that overall abundance, so the ddPCR copy number per leaf is lower. And this shouldn't surprise you, these phages are obligate killers, but it is at least suggesting that they really are finding hosts and knocking them out in this community. That effect on density goes away after day 7, although a lot changes on the leaves, carrying capacity is reached, so probably there's a lot of strain switching here as the community is establishing and stabilizing. The second thing we can do then is sequence our microbial community and ask what effect phages had on the whole community composition. And I'll just show you one example of that here to say that, again, when you have phages present, there's a clear compositional shift. So this is a PCOA plot where we're just looking at community composition and beta dissimilarity among them. So all you need to know here is if two points are further apart, that means the community members have less in common. So the community is more different. And so again, you can see that the bacteria and phage replicates cluster out, whether it be day 1 or 7, and the bacteria only cluster out. And the treatment, whether or not phage is there or not, explains 13% of the variation in community composition. And so again, this just suggests that if you're trying to explain microbiome diversity, whether it be a microbiome in a human gut or in a lake, ignoring the phages, you're going to miss this sort of explanatory power of at least 13%. And then the final thing she was able to do using this same data set here, the 16S amplicon data set, is ask what impacts phages had overall on diversity. And I've already given you a theoretical reason why we expect phages to maintain diversity. I've already shown you that this happens in a test tube. So hopefully this won't come as a surprise, although to me, science is always surprising when it meets expectation. And so what you're looking at here is on the left, alpha diversity. So you can think about this as how many OTU or what the richness of species are there. And at day 7, so we don't really see an impact at day 1, which is not particularly surprising, but at day 7, once these communities have established, we find that indeed when phages are present, there's a higher diversity of bacteria in these microbiomes. And so that fits beautifully with theory. The more surprising result for us, although again does fit with theory, is on the right here. And what you're looking at is the average Bray-Curtis distances among replicates. And so another way of saying that is how different is the microbiome of 2 plants that received only bacteria from one another, or 2 plants that received bacteria and phage from one another. So within treatment, but differences among individual plants. And what you can see here is no significant differences at day 1, but at day 7, we actually have decreased beta diversity in the presence of phage. And I think this is really neat because it's suggesting that phages are really moving the microbiome in the same direction. So they're sort of reining in the microbiome in a similar way. Okay, so I'm just going to finish up here in the last 7 minutes or so by zooming in a bit on single pairwise interactions. And so, as I've already mentioned, we work primarily on Pseudomonas syringae, a plant pathogen of broad host range, and we focus on tomato plants. And this work here is a recent paper that's come out from Kathy Hernandez. And so the citation's down there if you're interested in the full story. But we became very interested in the idea that coevolution is likely to be very different in a test tube from in a plant, and I've already shown you some evidence that that's true. But we wanted a very specific test of this. And so because we know that we can grow our strains on a tomato plant in very controlled conditions, we thought this is a nice comparison to a test tube. So what I'm showing you here is our in vitro lines. This is a typical experimental evolution setup where on the far left, the bottom here, the treatments, we have bacteria alone. And the passage 3 is the midpoint, passage 6 is the end of the experiment, and you just look at whether they evolved resistance or not. And the bacteria alone lines do not evolve resistance. And the co-evolved, or if you allow either of them to evolve as a fixed against the other one, so in other words, phage can evolve to fix bacteria, or bacteria can evolve to fix phage. In either of those cases, we see that resistance spreads. And the 2 different colored dots, it's not really important, but that's ancestral phage or the contemporary phage during coevolution or evolution. So in a test tube, resistance evolves very quickly is the take-home message from this slide. And that's not surprising. What you can then do is take resistant mutants and susceptible mutants, put them back in a plant in the presence or absence of phage, and measure selection for that type. And I'm just focusing here on the 24 hours. So this is, you inoculate the plants and 24 hours later, you can use droplet digital PCR to measure Pseudomonas syringae. And what I want you to take away, again, this is in a test tube, is when there are phage present in that tube, the bacterial density of Pseudomonas syringae is lower for susceptible mutants than resistant mutants. In other words, selection should favor resistant mutants. And indeed, that's what we see. Resistance spreads in a test tube very quickly. In most cases, we see it happening in under 48 hours. Okay, so what happens in a plant? In this case, she took the same strains and the same experimental setup, but now put these bacteria and phage in a plant. And I'm showing you one example here, but she reran the experiment in many different ways and many different MOIs. And what we found was that we did not see the evolution of resistance. And this, importantly, and in contrast to what I've just shown you from a natural microbial community, this is resistance by mutation. Whereas previously, those resistance mechanisms were open to strain replacement, species sorting, etc. If you're looking at evolution by mutation in this particular system, it seems like we don't see the evolution of resistance, whereas we did rapidly in a test tube. Okay, so this blank figure should be pretty convincing that it does not happen rapidly, but why? And so we took that same approach that I just showed you, where we took our resistant mutants, and importantly, these are resistant mutants that were evolved in vitro because we don't find them in planta, but we can still put them in planta and ask how they grow in the presence or absence of phage. And in total contrast to the figure I showed you previously from in vitro, in this case, in the presence of phage, there is no selective advantage to resistance. Okay, so that's not to say that phages aren't doing anything, because you can see if you just look at the susceptible mutants that their growth rate is decreased in the presence of phage relative to the absence if you compare the blue and the green bars there. So phages are killing their hosts in this environment, and they're not killing the resistant mutants. But the difference here is that that strength of selection is not strong enough to overcome the costs of resistance. And we've previously demonstrated costs of resistance in this system. The majority of resistance mutations happen via changes in the LPS pathway. And we know that the LPS receptor that these phages use to bind, changes in that receptor can come at huge fitness costs, especially in the plant environment. And that was work previously shown by Sean Needham in my group. So the takeaway from this section is that bacteriophage coevolution does matter, but the impact of it is going to vary depending on the environment. And we need to be a little bit cautious moving from test tube experiments to making predictions about the real world. But you will notice that there's this potential juxtaposition between what I've shown happens rapidly in a natural system and what's happening in this more controlled system. And I think there remains a lot of work to be done as to how much of what we see in terms of phage-mediated selection in nature is the result of mutational change within a lineage versus larger ecological processes. And so to summarize, hopefully what I've been able to convince you of is if we're trying to understand microbial communities, which as a field we are very interested in at the moment, we know that these communities are complex in terms of diversity and across space. They're very highly variable, so individuals host different microbiomes, different lakes, et cetera, have different microbiomes. They're dynamic across time, and if you're thinking about a host-associated microbiome, they're critical to shaping host phenotype. And through the series of experiments I've just shown you, hopefully I've made a convincing argument that phages are likely to be important in all of these, and through this coevolutionary process, they're likely to shape composition, chain by adapting to local bacteria, potentially driving different populations apart, or in some cases moving them together, as I showed you from the tomato plants, drive changes in composition over time, and as I showed you in terms of prophages in particular, be important in shaping their bacterial host phenotype, which could have important implications for the eukaryotic host, again, if it's a host-associated microbe. And with that, I will thank my group, the organizers of today's and tomorrow's workshop, and I'll take any questions.
Thanks for that, Brett. That was great. I just wanted to open up if anyone had a question before I did.
I'll ask the first question again. Hi, Brett, this is Sean.
Hi.
How quickly are the bacteria multiplying in planta? Because you've got a difference in speed per unit of human measured time. Can we adjust it at all to bacterial generation time? Thank you.
Oh, I would love to know that answer. Well, we can get at it a little bit with our droplet digital PCR now. We can look at growth rates. And it's a good question. You know, I wonder, we could probably mine Kathy's data set a bit better to get an idea of that. But we're still measuring generally our droplet digital PCR. We're measuring after 24 hours, 48 hours, 72 hours. I think we would need a much more fine scale resolution to not also then be averaging over death and so forth. But it's a good question, and I think we could potentially do it with this approach.
Perfect. I'll ask the next question. I was going back to the tomato plants and viruses on them versus viruses added to them. This kind of allochthonous versus autochthonous approach. You know, in California, you can go several months with no rain. And this idea of watering, we're watering plants. So if you think we're watering plants for the whole summer and we're adding a certain water source, and then they maybe get used to those phages or those viruses in the water source, but then we'll get rainfall probably from the ocean that comes in, and we're adding these new types of viruses. So do you see impacts maybe, or have you thought about that, maybe different water sources and this idea of the community switches with the season because of this?
I think it's a great question. I haven't thought about it within an agricultural setting, to be honest, especially irrigation water, but I think that's a really nice place to look. Where we are doing that is, I mentioned briefly, fire blight disease. We've been sampling pear trees for 3 years now, monthly in Berkeley. And we should be able to have, once we do the metagenomics of that, we should be able to get an idea of whether we see wholesale changes in the virome that's there, depending on rainfall. And I think that would be really neat, but unfortunately we're probably at least a year from that answer. But I haven't thought about doing it in an agricultural field, and I think that's a really nice idea.
I've actually got a follow-up question on that. So Britt, just thinking about it from generalist and specialist viruses, and so leading on from the previous question, to me, if you've got different sources, I would think that the hosts are susceptible to different varieties of viruses. They're not very unique. So that means the viruses, the phages that are infecting them are not specialists, but they're more generalists.
So again, that's a huge goal of our PAIR project, is to see whether that host range changes over the course of the season. But something just occurred to me that's relevant to both of these questions, which is that if we're thinking about this dispersal and immigration and turnover as a function of rain, for example, one of the critical and I think unanswered questions that I would love to hear from this community, if there's data out there that I don't know about, is the relative dispersal rates in the environment of phage versus bacteria, because I think that's critical. If we're looking in rain, you know, how much, of course, we know Pseudomonas syringae, our focal pathogen, moves through the water cycle, but we know much less about phage movement through the water cycle. And so I would be very keen to, if anyone knows of good data sets, contrasting movement of phage and bacteria. We might not have time now, but please email me if you do have that. I would love to have that conversation a bit more. Or in marine systems, for that matter.
Let's thank Britt again. I think this might be a great transition to Rachel, so thanks.
Hi, this is Rachel. Did you want me to just start and share my screen, or how does that go?
Yeah, can you start your camera first so we can see you? There you go. Yeah, and then click the share on the button and share your screen, and we should be good to go.
So I should have my slides in like view mode or whatever?
Okay, we're not seeing your screen share yet, but yeah, if you can go to the slideshow mode, and then we'll see them full screen. Yeah, we're seeing your desktop now.
Yeah, so now I have to switch to...
Yeah, on top, there's a slideshow menu.
I don't know why I can't, sorry. There.
Yep.
Is that
we're
okay?
Good to go.
Perfect.
Yep, go ahead.
Recognize that, Ken? Okay. Okay, so I'm sorry I haven't gotten to participate in the whole thing of this workshop. It seems really cool, and it's very awkward for me to be presenting without having anybody to talk to. I assume there's people out there, and I'm happy to take questions whenever, but I know that's kind of awkward in this scenario. So I guess I'll just talk through the way we're looking at viruses now, which is a little bit different than I think probably, I know at least the way Britt was describing the ecology and evolution, and I think that it's a little bit different than the way people think about it in microbes normally. So we're thinking about viral symbiosis. And I wanted to kind of go back all the way to the beginning because I'm really interested in the origin of life. And we were in this NASA Astrobiology Institute that was looking at the processes of the origin of life. So I just want to remind you a couple of things about that and then put viruses in that context. So, of course, you know this 3-domain tree of life that was published by Carl Woese in 1977. And I'm almost, embarrassed to put it up there again, because I hope we all take this now as a true understanding of the way life emerged, even if we're not sure exactly how the archaea and eukaryotes are related as the trees are changing. But the same year, he also published a second paper, which was on the concept of cellular evolution. And people know less about that work, but I think it's really, really important. It was less about the relationships between the organisms and more about the process of evolution and how that changed in tempo and mode over the course of evolution. So very quickly, what he said was that we know that there's fossils of cyanobacteria, and I know these dates change around, but it's something like 3.5 billion years ago. And by that time then, if we believe those fossils and those dates, we would have had an entire cell that was functioning and doing oxygenic photosynthesis, which is a long way to go if the Earth started in 4.5 billion years ago. So in that first billion years, we went all the way from chemistry to full cell biology and oxygenic photosynthesis. Whereas since then, really, in comparison, we've done very little. We've just kind of articulated on these things, and we still kind of function basically as cells in the same way. So his question here is what changed and what was different about the pre-cellular part of life and the last 3.5 billion years. And his idea was that there was a change in evolutionary mode. And he called this change in evolutionary mode the Darwinian transition, because he thought one of the things that made this transition happen was that the life changed from the form of life he calls a progenote, which is not a cell, to a cellular form of life. And this mode of evolution that changes from this progenote state to a cellular state is what he called the Darwinian transition. So let's think about what evolution is like in a progenote state where there aren't cells that are defining an individual. And the cost of that, or how that changed the evolutionary mode. He obviously thought that it slowed things down, and that kind of makes sense when you think about the way horizontal gene transfer and recombination shapes the way cells evolve today. So the evolutionary mode of the progenote is one that he thought was getting the relationship, was just trying to put together the relationship between genotype and phenotype. So there were no cells. It was this kind of supermolecular aggregate that kind of was evolving as a pool. And that actually, in classical population genetics, allows selection to work more quickly because it can work independently on single pieces, single components, and optimize them specifically for the specific conditions that the progenote is evolving in. And it also allows new components to come together rather freely without the constraints of having to interact or be linked together. But it comes at a cost because it homogenizes the way selection acts, and you lose diversity in that process. And so this kind of prevents the specific evolution of a single context-specific interaction between these molecules. So he kind of, I think, based some of these ideas on Saul Spiegelman, who was thinking about the origin of cells as well, and thinking about why chromosomes kind of consolidated at this Darwinian threshold. And so he thinks that, you know, this complexity and the size of the genome was increasing and possibly changing into a DNA genome for this reason, to be able to have more complexity and be able to solve more complicated problems. But putting it together, again, has this trade-off, because now you have everything linked together into one chromosome inside one cell, and all of those things being linked prevents selection from independently acting on any one. So this may be going a little far, but I'm just reminding you of population genetics that I hope you had a long time ago, where if you have high linkage and the selection acts on a particular gene, it's going to act on the whole region of that. And in clonal organisms, that's the whole chromosome. Whereas if there's little pieces and aggregates that are independent, selection can act on each one of them independently. So it decreases the efficiency of selection, but increases the specificity of selection to consolidate genomes through this Darwinian transition. Okay, so in that context now, we can start to think about viruses and what we know about the way these consolidated chromosomes all together are evolving. And I think that we all know that by comparative genomics with many different strains and population genomics, that there are still a lot of mobile genes that are moving around. So we haven't fully crossed this threshold into completely vertical transmission. Instead, there's components of every genome that are coming and going that have not passed this threshold. And they allow selection to act efficiently and more specifically on those components than otherwise might happen. Now, the difference here is that these are independent genetic units. Most of the time, I think they're viruses and other mobile elements that then get kind of co-opted by their host. And so this is where the pangenome concept comes in, which comes from looking at many different microbial genomes and seeing that there's a core genome that evolves as you would think it does by the rules of population genetics and the modern synthesis. And then there's this variable genome, which is the set of genes that come and go that we're still trying to figure out the rules on. And those are important genes that encode really important traits. We know this mostly in pathogens where we've seen toxins, virulence factors, and antibiotic resistance moving around and spreading on these highly mobile genes. That makes sense in the context of selection. These things are more efficiently under selection if they're highly mobile. So, but if they're their own lifestyle, their own fitness, viruses, then we have to start thinking about the interaction between the chromosome and viruses as a symbiosis and how they evolve independently and separately in this mode of evolution that I think is kind of beyond the Darwinian transition that brings us into this more mobile world. So what are the evolutionary rules? We need to start looking at the different types of viruses there are. And I know that viruses are very famous for their antagonistic effects on communities and populations. And they certainly are changing dynamics in microbiomes and are really important in that way. But I think what's often missed is the other forms of viruses, the ones that are latent that are in the chromosome as proviruses and are being transmitted from generation to generation vertically and co-evolving with their host chromosome background. And then chronic viruses, like the one I'm going to talk about, which like SSV, which doesn't kill its host and kill its host as it is transmitted, but buds from the host. And so that allows this symbiosis to possibly evolve towards more of a mutualism between the virus and the host chromosome. So I think we know very little about viruses in general and their different lifestyles because they're really hard to study on kind of a population level. And one of the breakthroughs that is allowing us to do that is CRISPR-Cas immunity. So I don't need to talk about the specifics of CRISPR-Cas immunity and the elegant immune system that has evolved, because I'm sure you all know about that. But I just want to remind you that this also provides a tool for evolutionary biologists to keep track of virus-host interactions over time. So that in the process of acquiring immunity to viruses through CRISPRs, bacterial and archaeal cells add new spacers onto into their genome, add a new component, a small component, sequence, piece of the genome of the virus into their own chromosome. They record a memory of that interaction in the CRISPR-Cas immunity. And that allows us to look back at populations and try to figure out what kind of interactions are happening between viruses and hosts and how this might be impacting their evolution over time because of the population signatures that are in the CRISPR arrays, that are in the immune diversity. So we've been using immune diversity to link microbial cells to their viruses, and also to try to understand how their interactions are happening in natural populations, because this is really hard to do. So now I was just going to give a little example of what we've learned by doing this and how this uncovers aspects of symbiosis that you might not have thought about, or that one might not think about in any system. And that is to look at this population of Sulfolobus icelandicus. Sulfolobus lives in geothermal hot springs in metapopulations, and it is a hyperthermophilic crenarchaea, and acidophile. And because it evolves in these metapopulations, we're looking at dynamics across this like nested scale, which is how the virus interacts with the host, with its host, with Sulfolobus, how that interaction changes interactions with other organisms in the hot spring. And I call this an island because it's very clearly defined, and so we can link dynamics that are happening between virus and host to this one relatively well-defined ecosystem, and compare the different ecosystems to each other and to understand the dynamics that are specific within and between them. And then we're putting that into kind of a whole big global context, although we don't see migration between these different locations that we've sampled for viruses or for hosts. So we've looked really closely at two populations of Sulfolobus, one from Yellowstone National Park and one from Kamchatka, Russia, and kind of looked at the dynamics that are happening in those populations in their genomes. And what it told us was that the dynamics are basically driven by viruses and mobile elements. The telling us means that the most variable components of the chromosome are these elements coming and going, and the factors like CRISPRs that mediate the interaction between viruses and their hosts. So we've isolated a 10-year time series of hosts and viruses from these two independent populations. And you can see here that they're different between the different populations. So in Yellowstone, we find mostly SIRVs, which are lytic viruses, while we also find chronic SSVs as well, but we don't find them plaquing as much, and we find them integrated into the chromosome. In Kamchatka, we find SSVs that cause plaques and no SIRVs, but we find a couple other lytic viruses that we're working on characterizing. So we have the same interaction between a single host type, Sophilobus type, and the same virus in two kind of different ecosystems with other viruses around them. So we're trying to understand how the coevolution between the chronic virus and its Sophilobus host is changing in these different viral contexts. Okay, so lots of years of work by, mostly not done by me, have characterized the interactions between Sophilobus and SIRV, and also between Sophilobus, spindle-shaped virus, and Sophilobus. And we know that this SIRV is lytic. It has this amazing pyramids that cause the cells to lyse, whereas SSV is a budding virus that is chronic. We know that that both the host and the viruses are globally isolated. So there's specific genotypes in Kamchatka that are different from the ones that are in Yellowstone, suggesting there might be local adaptation between hosts and viruses in these contexts. And we've looked most closely at the SSV and Sophilobus islandicus interaction in Kamchatka, Russia, which is a large population that's well mixed and pretty stable over time, meaning we've gone there in 2000 and 2010 and seen the same level of CRISPR diversity and virus diversity maintained over time. And the maintenance of that large diversity of CRISPR alleles tells us that there's not like selective sweeps of viruses that are happening in these populations that are clearing diversity, at least immune diversity, out. So that's an interesting dynamic that we think is conferred by CRISPRs. So the CRISPRs kind of allow this stable diversity in CRISPR in the hosts to evolve. And we've looked very closely at this using modeling approaches to identify what aspects of the system might allow it to evolve this diversity, and where, if we don't have this diversity, what the impact on the host and virus population are. And so this is an example, an old example now. We have many more complex examples that we've looked at more closely now, but it's a simulation showing that this kind of diversity can persist. You can see there's many colors of host in some of these peaks, and those colors mean that you have many different CRISPR alleles coexisting in a single population. And they all match the same virus, but in different ways. So it's the idea that you can have a one-to-many, one virus matches many different immune alleles that controls the dynamics of this system. And so we've looked at what those dynamics are, and we see that when there's a high diversity, or what we call distributed immunity, the viruses have difficulty persisting and go extinct in the population, probably because they're at very low numbers, because they have to find a host. And so we see this in Kamchatka. We see that there's these SSVs that are persisting, and yet we see high distributed immunity to this virus in this population. And we were asking how this could be, because the virus shouldn't be able to survive and persist in an environment where there's this really diversified CRISPR-Cas immunity. So we've calculated population distributed immunity in the different populations that we've looked at relative to SSV, and seen that it's pretty high, and that if you use these calculations to try to predict whether the virus should persist, it's very unlikely to invade and persist in this ecosystem. It's just really hard for any SSV to find a host it can infect. So we tried to look at this in the lab. We took SSV, the SSV9, which Ken isolated from Kamchatka, Russia, and we were able to infect our host with that and establish a chronic infection in the lab. And we can see that the infected cells grow, as you would predict with a chronic virus, and produce viruses over time. But also we've been able to show that CRISPR immunity works against this virus by genetically engineering the system to knock out CRISPRs, and show that only in the CRISPR knockouts can this virus persist. So there is this dynamic where CRISPRs recognize SSV, and yet SSV persists in the population. So we had this question of how this can happen, and we tested this question by competing an infected strain against an immune strain in the lab. And this is where we uncovered something that we were kind of surprised to find. That SSV infection has a small cost to the host. So the persistent chronic infection has a small cost to the host. We haven't been able to measure a cost in the of the immunity. But when we put the two cells together, the infected strain wins every time. So the top graph here, you see the competition between an immune strain and an uninfected strain of sulfolobus, and you can see that they persist over the time scale that we were looking at. Whereas if you put the same background infected strain and compete it with an immune strain, the infected strain wins and actually kills off the rest of the immune and uninfected cells. So we've looked at this as, we think this is a mutualism. It allows a host to invade even when rare. The host cell, if it's carrying a virus, it can invade a new population by killing off competitors whether or not they're immune. And that helps the host and it helps the virus, because the virus probably wouldn't be able to find any hosts in that very diverse CRISPR population. So we have this idea that the chronic virus has turned into this mutualist, conferring a very adaptive trait, in a competitive trait to the host, which is killing its competitors, and in the meanwhile also establishing its own fitness in a population that has very few susceptible hosts. So this is just an example. It's, you know, an idiosyncrasy perhaps, but I think it shows how the virus-host interaction can evolve new traits for the host that change its fitness in a population, and that is the kind of selection we're talking about when we're talking about moving genes around. And sometimes virus infection really changes the traits of the host as it's evolving through evolutionary time. So just don't forget, I guess it goes without saying here that these types of interactions are happening everywhere all the time. And maybe they're compensating for this loss of flexibility that came with the Darwinian transition, where we transitioned to an individual cellular vertical mode of evolution. So this is my lab. I chose two different pictures that are actually out of date now, but because the work has been going forward through time to highlight the people that actually did the work. In the top picture, Maria Bautista really isolated the SSV and made it tractable to work with in the lab in our model system. Changyi Zhang did all the genetics that we needed to test CRISPR function, and Samantha DeWerf has been working on the interaction between infected and uninfected cells. And we are very collaborative with lots of labs all around the world, and that's really important to note, especially on this work. We have worked with Joshua Weitz to do the modeling and Mark Young to do the virus isolation, and we're now working on new models with Mercedes Pascal at the University of Chicago. And so I'd love to have any questions if that wasn't too fast.
I guess let's open it up for questions if anyone has any.
I have far too many, but I'll wait. Well, maybe I'll start with one just to fire things off. The killer archaeas, or the sharkeas, I saw your nice little image there in the corner. Do you think that this killer effect might also have something to do with what Maria found, again, with the SSV9 and the dormancy effect. Do you think there might be something there? Do you have any ideas what might be going on there?
Yes, and we're working on this. It is the same. We've identified the killer as a toxin. It's a protein that's produced by the cells. It's exogenous. It can put the cells into dormancy if it's washed away, but if it's not washed away and persists in the environment, then the cells die. So I think what the fact that she was seeing with dormancy was that she would challenge the cells with the supernatant, presumably with the toxin in it, and then wash it away. And so the dormancy was a reaction to that, to seeing that toxin and then being released from whatever effect it was. We're working on what happens in the cell in response to this toxin and why the infected cells don't get killed by the toxin themselves and are resistant to it. And we don't have the answers yet.
So if no one else will, I'll keep going on this too. So there, as you well know, there are toxin antitoxin systems also in sulfolobus, but many other organisms. Do you think this is a more generalizable thing? You can start thinking about toxin antitoxins as well in these sort of not necessarily killer archaea, but killer bacteria, killer other kinds of things.
Sure. And I also think that it's actually good. So one of the puzzling pieces for a long time for us was why would a virus want to kill possible susceptible new hosts? And I think that we think now that the antitoxin must be encoded on the virus. So if the virus can get in, is not recognized by immunity, then those hosts live. So it's kind of a way of the virus screening through immune cells because it's like a addiction system, but exogenous, right? So you have to get infected in order to resist the toxin, which I think is really interesting and seems pretty powerful. So I would be surprised if it hadn't evolved elsewhere.
Very cool. I could keep asking questions about this for ages. As you know, I'm a bit of a sulfolobophile. But okay. In the interests of everyone, needing physiology breaks and so on and so forth, and this is, I think, a great lead-in to Joshua Weitz, who's actually talking right after the break.
Oh, great.
Unless somebody else has any other thoughts, should we just go ahead and go on break until 10:40 Pacific? Does that work for everyone?
I think that's a great idea. I also wanted to highlight, if everyone has time, actually look at the comments that are in Zoom. There are actually some great engaging conversations and be great to get more feedback from people or just really think about it. I really love the discussion that's going on there.
Also, we will have that transcript available. It takes maybe a couple of days for us to figure out how to do that. But we are recording everybody's comments. So a lot of these topics can be folded into our follow-on discussions, in the process of putting together a paper and the white paper. All right, break time.
Thank you.
We're good to go.
Okay, great.
Thank you.
Thanks for having me here today at this virtual workshop. My name is Joshua Weitz, and I'll be talking about viral infection modes and invasion fitness across a continuum from lysis to latency. And I've hidden myself, so I assume you can see me and you can see these slides. Is that right? And you can hear my voice. Someone get great. Okay, and let's see, as long as I can get it to advance, that would be good. So we've heard a lot about viruses already today, I'm sure tomorrow as well. And usually when one thinks about viruses, at least in a public setting, it's often because one is thinking about, let's see again if my buttons are clicking here. Sorry for it made one click. Let me see if we've try that again. There we go. We usually talk about viruses in public context in terms of those viruses that might cause mortality and morbidity with respect to humans, human diseases like Ebola, Zika, or influenza. And I think it's fair to say that this audience knows that viruses infect organisms really across the span of diversity of life, from humans, mammals, birds, insects, also plants, but crucially also microbes. In other words, microbes get sick too. And I've given some examples, and the prior talk touched on one of those, the sulfolobus spindle viruses and other archaeal viruses. And also, of course, there are viruses of bacteria, amoeba, and so on. And so I'm going to spend some time today using these viruses of microbes, particularly viruses of bacteria, as a means to think about the ways in which infection mode might differ and not necessarily always be antagonistic to hosts. So just to get everyone on the same page, you're probably aware that when bacterial viruses phage or bacteriophage are in the environment, they might diffuse until they're in contact with a target cell, inject their genetic material into the host, redirect cellular machinery in order to make more genetic material, viral genetic material, which can self-assembles and into these mature virions, and through a time process, often there's a hole that's made in the inner membrane, some cleavage in the cell wall, and then through a time process releases these virus particles back into the environment. And so clearly for that particular cell, that's the end of its life. But that doesn't necessarily mean that the death of one cell is the death of a population. So because of this antagonistic relationship, the fact that this virus in some ways consumes or eats the host, phagos to devour, the predator-prey model has been the basis for quite a lot of studies of virus micropopulation dynamics, how we move from cellular level models up to population level models. And so here on the left, I've just given one variant of a classic example. And again, my background is in theory and modeling. And here you have interaction between some resources that are being flowed into the system, cells that take up those resources, and viruses that infect, reproduce, and kill those cells. And the left-hand side denotes a nonlinear dynamical system where you have resources R, bacteria N, and viruses V. And again, the process of nutrient consumption turns resources into cells, infection turns cells into more viruses. And with these simple ingredients, one can track the expected dynamics in time. And here on the right, I've shown time implicitly, where prey is on the x-axis and predator is on the y-axis. And what you see are these counterclockwise cycles. In other words, when there are a lot of prey around, the viruses do well, increasing in abundance. When there are a lot of viruses, they drive the prey down. When there are few prey, then viruses go down. And because there are few viruses, then bacteria can recover. And these are really Locke-Volterra-like dynamics. So for the same reasons you see these oscillations within Link's hair systems, it's presumably the same reason you might see them within virus microbe systems. And there's experimental evidence that these kinds of population cycles can be observed in the laboratory. These are classic work, Brendan Bohand and Rich Lenski, and they're predator-prey-like cycles between phage T4 and E. coli B. It's about a 7 to 8 day experiment here. And you can see with time, oscillations of large magnitude in the population densities of viruses and hosts. You can see at times virus to host ratios don't just get to be 1 or 10 to 1, but can be 100 to 1 or more. And the oscillations are not because of some exogenous driving of the system. It's not as if the system is being driven by some oscillatory research input. This is in a chemostat with steady flow of minimal media, and nonetheless, the feedback between virus and microbes at the cellular level translates into endogenous large-scale oscillations, and they're shifted. If you notice, the peaks of the viral's abundances are slightly shifted forward in time with respect to bacterial peaks. Okay, and so with that in mind, and I'm sorry that there's some technical issues here, the sharing screen seems to forget that I am here with a computer. So maybe let me go back. Is there any way I can do this just by zooming in? It's the sharing of the screen seems to be bad. Let's see if I go into that view. Can you still see that? Okay, sorry about that delay. This way, at least I can click on the new slide if I need to. And so these virus-host interactions modify the fate of cells on short time scales, but on longer time scales, we might accept there to be oscillatory dynamics. And then once you start scaling this up to ecosystems, this effect of killing individual cells and populations can have large ecosystem level effects. Particularly, we're interested often in global oceans. And part of the reason for that is that the number of viruses outnumber those of microbes largely bacteria, but some archaea, anywhere from about 1:1 to 100:1. And if you look on the axes here, we're talking about typical densities of 50 million viruses or virus-like particles per milliliter, again, in excess of those of bacteria. So all that lysis presumably leads to big changes in microbial densities, effects on microbial mortality, as well as the regeneration of carbon and other nutrients back into the environment. But all of the examples I've given thus far really focuses on the effect of viruses as killing agents, antagonistic, obligate intracellular parasites that end up just killing the host in order to make more of themselves. So the question I want to ask and really focus on in my time today is, do viruses and microbes do more than kill or prepare to kill? And the answer is yes, and has been known to be yes for quite a long time. And lysogeny and temperate phage really provide a number of lessons from a simple system, going all the way back to Andrei Lwoff and others of the pantheon of those who understood viruses and tried to understand the viral impacts on gene regulation and molecular biology. And the key idea within temperate phage is that you have the injection of the genetic material into the host, but then a genetic switch, really a decision between two pathways. One, which is lysis, which I've already described, and the other, which is integration of the viral genome into the cellular genome, where it is termed a prophage. And that prophage, that viral genome, can then heritably be passed on from mother to daughter cell across the replication, and at some later point can be induced. And by induced, I mean that the prophage excises itself from the cellular genome, reinitiating the lytic cycle, and then heads out again. And although this is classic work, there's still quite a lot of activity trying to understand these switches, both that switch in, like how to be temperate, how to make this decision after the virus injects its genetic material, and then how the process of induction works. This is one example from the work of Ido Golding, who is soon to be, I believe, at UIUC, has been at Baylor College of Medicine for a number of years, and using a labeling system is able, you see in panel B, identify when a phage, because the capsid has been marked with this YFP, has attached, to a cell, and then depending on the fate, there's a reporter system that those cells that have been fated to lysogeny turn red, so they're normally growing, and you can see them actually divide, and that red color pass heritably between the two cells, and those cells that then produce new phages produce these, again, labeled phage. And the point I want to make here, although these are dichotomous outcomes, these decisions are probabilistic, meaning it's not as if a particular cell or particular virus encodes, yes, 100% of the time going this way, and then deterministically, depending on the state of the host, it'll pop out. There is a relationship between cell state, particularly if the cell is not in a good state, maybe a phage can detect that and get out. There's also a relationship between multiplicity of infection, other factors, and that is a long and interesting topic, maybe for another day. And all that revolves on how to be temperate. How does a virus regulate, detect what's going on in a cell in a way that can change these probabilities, this initial decision switch and this induction switch? But there's another question, which is an evolutionary question, which is why to be temperate in the first place? And Frank Stewart and Bruce Levin in the mid-'80s asked this question, and they proposed one of many, a feast or famine hypothesis. The idea that temperate phage do better when there are few hosts that are available, and the extracellular mortality rate is high, but they did offer a caveat. And they said, in spite of the intuitive appeal of this low-density hypothesis, they're unable to find solutions, meaning they could impose those conditions, and that seemed to favor the temperate phage, but it wasn't as if they understood the principles by which that might happen. So the idea is that maybe when there are fewer phages around, it's better not to kill off the last hosts, but that has a bit of a group selection or aspirational level to the argument. Okay. Though there is some evidence that this is really what happens, or at least that had been the paradigm. In marine systems, there was a notion of seasonal time bombs, the idea that in low productivity, low density environments, this is work by Jennifer Brum and colleagues, there are other similar studies, that when you look and use an induction assay to try to pull out the number of lysogens, pull phage out that are lysogens, when over the winter or spring there are less productivity and fewer cells, then in fact you see a lot of lysogens, and in the summer when things are productive and a lot of cells, then you don't see many lysogens. In contrast, when you look under the microscope, there's a higher fraction of lytically infected cells, okay? And so that's the contrast here between the top and the bottom panel. And this had been the paradigm until I would say just a few years ago, when an alternative hypothesis was presented. It's termed piggyback the winner. Some of you may be familiar with it. The idea is really quite the opposite. The claim is that lysogeny is positively correlated with increases in host density and productivity. In other words, lytic activity goes down as there's more productive or denser environments. And there's a background to why this was of interest to the authors, and also some additional context on density relationships. But there's some direct evidence that they presented by looking at a range of microbial densities, and on the y-axis here, evidence from metagenomics that there was some increase in lysogeny. We have to use proxies here. By looking in viromes, they look for things like the percent of provirus-like reads, things that might indicate the potential for latency. And despite claims in the paper, I would argue, as you see here, that there's not a really strong relationship between the y and the x-axis for provirus-like reads. There's not a strong relationship between the y and x-axis for integrases, nor for excisionases. And these are all indicators of things that look like they have the potential getting in and getting out. So the takeaway, at least from my perspective, is that at the moment there's an absence of evidence for a positive correlation between lysogeny proxies and cell density. And one can read more about that. There's other ways to look for these proxies by looking directly at ratios of viruses to microbes as a proxy. I think there are other issues there. But nonetheless, I do think that this paper, and to some of this debate and back and forth, has raised perhaps even a more important question, which is, what are the environmental conditions that should favor lysogeny over lysis? I don't think that question has been resolved. And I'm going to try to begin to address that question today by turning to an old lesson that some of you probably know, and turns out that there are different ways of saying this in different languages and cultures. The one I'm familiar with is a bird in the hand is worth two in the bush. The idea you have something now, and you might trade it for something in the future, but you better trade up, because you're not certain of how much you're going to get. And so I claim that this relates to a puzzle that we should be asking, which is, a virus in the cell is worth N in the bloom. But what is N? Right? The virus has the cell right now. Sure, it could put out a whole bunch of virus or virus particles back into the environment, but how many is worth sending back out versus the one cell that you have right now? And so I want to make this comparison more direct by talking about the notion of viral proliferation and thinking about what really is the fitness of these different viral strategies when we view them at the individual level. And I'll start with the typical idea of lytic viruses, where you have this virus, it encounters a cell, infects the cell. I'm going to call that the mother virus, as opposed to the mother cell. And it eventually will lyse and produce 100 virus particles. I'm going to make the claim that that is not its fitness. 100 is not how many offspring that particular mother virus has, because 97 of those virus particles might never find a new susceptible cell. They might decay, be unstable, or so on. And in this particular example, only 3 encounter, and successfully infect into a new cell, and we'll call those daughter viruses. And so therefore, I would claim that in this case, the fitness at the individual level, the number of offspring over the course of the lifetime of this mother virus, is 3. And they've been obtained horizontally, via horizontal transmission from one cell to other cells in the environment. On the other hand, latent viruses also have a way of reproducing. The mother virus might divide and therefore produce progeny, daughters and daughter viruses, that they will count their own fitness on their own. And if the mother cell divides 3 times before it dies, whether because it's grazed or sticks to something or lyses for some other reason, now just through vertical transmission alone, it's also possible to have the same fitness. Okay? So the point I'm trying to make here is that 2 vastly different strategies can lead to the same notion or calculation of fitness at the individual level. And from an ecological perspective, it would be good to know how this depends on cell densities and other factors if we're to begin to understand which of these particular strategies might be favored, or a continuum of these strategies might be favored in the environment. So to do that, what my group has done in collaboration with Rachel Whitaker, with some folks in my group, Guan Li, a student, and Mike Cortez, a former postdoc now faculty, and Haria Gulbadak, also former postdoc now faculty, is take this individual perspective and translate it into a population perspective. So I've given you the example on the bottom left of what happens for the individual. The top is meant to be a box model describing susceptible cells that can become infected, that generate offspring, various virus particles, these viruses that then reinfect and start the cycle. And the right-hand side is the translation of these into a dynamical system. And I just want to point out here that this last equation, the change in the number of virus particles, goes up because of lysis. So every time at a rate eta that the cell is lysed, it produces beta offspring. And virus particles are lost because they're removed due to infection or due to decay. And I'm not going to go into all the details today, other than to say that one can calculate, instead of that number 3, a generic description of how many offspring the virus might have in terms of this complete cell cycle, from an infected cell to new infected cells. In terms of these details here, but I'll just translate them, which is the number of virus particles produced, you start with 100, a fraction of those reach a new cell before they decay, and only a certain fraction of them are going to lyse before they wash out. So that's the complete cycle. The virus gets into a cell, it has a probability of releasing beta particles, and only a fraction of those make it to the new cell, and that then becomes the horizontal fitness. And when one looks then at the relationship between susceptible populations, some ecological trait, and a viral trait, one finds that it's not inevitable that viruses can invade if there aren't enough hosts, or if their traits are not sufficiently favorable for that host. And there's a lot of examples of viruses infecting hosts that may not be the optimal host in the environment. Then you get the extinction or decay of that particular phage on that one host, whereas the better the virus is at exploiting the host, the more susceptible hosts there are, the easier it is for the virus to spread. And the takeaway here is that ecological conditions with more susceptible cells and viral traits that are more efficient in infection and also can survive longer in the environment could favor a lytic mode. Okay. Now, in contrast, we can do the same thing with the latent virus case, but here, rather than necessarily looking at the horizontal transmission, I'm going to focus on just the case where fitness is passed vertically. And again, I won't go through some of the mathematics here, other than to say that one can calculate this vertical fitness and reduce it down to this simple equation, which is how long the cell lives for multiplied by its division rate, which gives you the average number of offspring. And if that's greater than 1, then there are more daughter viruses than there are mother viruses, and therefore they are going to proliferate, et cetera, et cetera. So again here, this is an example as we increase susceptible populations. The red line is the horizontal fitness, and I've shown 3 blue lines, which shows how this vertical fitness changes with susceptible cell density. Notably, it goes down because if you infect a susceptible cell and there are many other competitors, you have to face all those competitors in the same niche. So essentially, there's increasing niche competition. Nonetheless, if there are benefits of lysogeny, in other words, if the fitness of the infected cell is better than that of the uninfected cell, then there's a larger domain in which, as you see, to presumably very high densities of cells, it actually might be better to be a lysogen than to kill the cell and try to find others. So the takeaway here is that ecological conditions with reduced niche competition, direct cell benefits, or low virion survivorship all favor latent strategies. And there's also things in between these dichotomous outcomes. So here's an example in which a cell is infected, and it can both divide, generating progeny that are themselves infected, but through a budding process rather than a lytic process, release progeny. And therefore, the chronic viral fitness is a combination between the horizontal fitness and the vertical fitness. And here in this particular example on the lower left, there's 1 division event and 2 of the budded off viruses that find new cells. So the total fitness remains 3, even though it's through distinct modes. And so the point I'm making here is that then when we think about chronic viruses and their fitness, it becomes an aggregation of both the horizontal and the vertical components. And the point I'll make here, I've already told you what those are based on, is that we increase susceptible densities. We can compare all 3 strategies, finding that maybe at lower cell densities, the vertical mode is dominant at very high cell densities, the horizontal mode, and there can be a mixed mode that can be dominating between, though the precise position of these can change. The more that the cell is benefited by the viral infection, that blue line can go up and start to move over to the right in cell densities. So again, the takeaway here is that temperate and chronic modes are favored when susceptible population is relatively low, lysogens have a relative growth advantage, or extracellular decay rates are high. So in fact, some of these early predictions by Stewart and Levin really are recapitulated, but here, rather than trying to figure out how to balance virus particles with infected cells, answering that question of how many in the bloom we should count, by calculating fitness in terms of these viral life cycles, we get a direct answer to this question. Okay? And you can read more in this virus evolution paper that came out earlier this year. So to answer this question, I think that we should be thinking about unified metrics and thinking maybe alternatively to the predator-prey paradigm to use an epidemiological paradigm, which is something referred to as the basic reproduction number. The average number of new infected cells produced by a single typical infected cell and its progeny virions in an otherwise susceptible population. Right? And using this perspective, we can, in an apples-to-apples way, compare both the horizontal mode, which produces virions but doesn't necessarily go vertically, and the latent mode, even if it doesn't make a virus particle, it still can have fitness in the sense of having newly infected cells. And the last thing I want to do here, and I've probably moved a little bit too quickly, as others have noted, it's hard to know what the feedback is, but I'm happy to take questions, which is in calculating these values of R naught, I've assumed dichotomous outcomes, either lysis or latency. It turns out technically it gets much harder, and I'd say conceptually gets much harder when there can be a mix between modes. And we know that phage lambda, temperate phage in general, latent viruses can go out of the latent mode and then initiate lysis, and it's been quite hard for us to figure out how to do these calculations until recently. So I'm going to now just in one slide highlight something that's on the bioRxiv, which is we've been able to come up, this is a graphical way of depicting what we think the right way to think about fitness is. And there's a lot of diagrams here. We actually use these diagrams to calculate. The idea of the top line is that this is a model in which E stands for exposed to the virus, I is actively infected via virion, and S is a susceptible cell, L is a lysogen. So P1, that first diagram says the cell is exposed, it goes infected, makes a virus, the virus contacts a new susceptible cell and makes a cycle. The black to black says a cycle is created horizontally. We call that a lytic loop. But we can also have a lysogenic loop, which there's a lysogen, it doubles. So the offspring is a lysogen, still is the virus, that's a lysogenic loop. But we can have something which is a lysolytic loop, which is the exposed cell integrates the prophage, comes out into an active infection, produces viruses, and goes back to E, but passes through the lysogenic loop. Those numbers then, we can calculate the fitness associated with each, we have to add them up. But it turns out when we do the calculations and to really understand them, once you start to mix loop, we have to think through 2 generations. But because we have to think through 2 generations, you can mix these modes. And there are all these diagrams here, and also explains why there's the power of 1/2 at the end. We have to discount the fact that we've looked over 2 generations, and we have to compare that back to the 1 generation case. This might seem a little complicated, though it's kind of appealing in certain ways. The advantage of it has been is that irrespective of the population dynamic model you write down, whether this exposed infected virus model, there's work by Thomas Berngruber recently on a resource implicit model with implicit infections, and the Stewart and Levin model, which is resource explicit. All of these models, it turns out, have the exact same formula, but the quantitative values differ depending on particular details. But these are the combinations of ways in which you can have these life cycles. And it turns out you can make predictions and they look more or less the same when viewed in this way. So the takeaway here is, if you're interested, I'm happy to talk more, and you can read more on the archive, that a loop-based approach decomposes viral fitness into lytic, lysogenic, and lysolytic loops. So we really have mechanisms that we can think about rather than just a long algebraic mess. we can think about ways in which these loops are contributing to viral fitness, which transcends model details, and I hope reveals some generic mechanisms for the benefits of latency. And then the last thing I'll ask is, what is a virus? I think this project in collaboration previously with Rachel Witter and Mark Young, and the ongoing work has, for me at least, raised some new questions, which is I used to think of a virus as a capsid containing genetic material, an obligate intracellular parasite. But then you might think, well, maybe it's the virocell, right? A lytically infected cell, or maybe it's a lysogen. And I would actually say now that it's really all of the above. The viral fitness in the environment depends on measuring the present and long-term value of infection, but we have to start thinking, I think, about the entire viral life cycle, whether it's inside or outside hosts. And I think it'll take some time to figure this out, whether through theory, experiments, as well as field work. And so with that, I'll wrap up. I think I just have a few minutes left. Again, acknowledging some of the folks here at Georgia Tech, collaborators, former postdocs, Michael Cortez and Hyria Gulbadak, who have their own groups in mathematics at Florida State and UL Lafayette. Also at Montana State University, Mark Young, and UIUC, Rachel Whitaker. If you want to read about the latest work, it's on the archive, and there's probably a lot more to see. We have a website, Twitter, and also code data information on the GitHub site. So thank you for your attention. Okay, I don't know if there's time left for, probably very little time left for questions, maybe just 1 or 2 minutes.
Yeah, we'll open up for one question.
Hey, Josh. Go for it. All right, thanks. Josh, I was just wondering about situations where the lysogen becomes inactivated during sort of one of these infection loops and how you might account for its fitness in that situation. Like, is it still a virus? Is it no longer a virus?
Yeah, so I mean, there's lots of examples of these defunct prophage, and that's one of the risks of staying in too long. And that certainly decreases if, even though we are counting fitness vertically, there is a presumption here that at some point the virus could still get out and disentangle its fate. And then when that stops happening, we can have that argument at what point that is, and maybe other people here are going to talk about it, that it stops being a virus and maybe becomes some other kind of genetic element. But at least in our case, that would be a loss because it's another way of losing the viral genome and its ability to have both routes at that point. And then the calculations we could modify, but that I would view not as positive for viral fitness. And Siobhan might have something to say about that.
I think this is a great pairing of talks, but I was actually thinking the same thing that Lewis was. Do you need to model the loss of intact viruses due to defunct prophages, or is that not important because you're not modeling inactive virions, defective virions in the model?
Yeah, so I mean, at least at this point, there would be probably another level to do, but within the lysogenic phase, there is a loss term. So I view that as an additional loss term. Whether or not that still confers immunity, is that cell now susceptible? Is it, maybe it's immune, but it can't transmit? We haven't gone into that level of detail, and I agree that that is a nice challenge to think about. But at least I think we should think of that as a loss in terms of the average fitness that it's expected to yield. Okay, I think we're at time. Thank you.
I guess we'll move on to you, Siobhan, now.
Okay, I've got Zoom open. I'm trying to share screen.
Okay.
I don't know if that worked. Am I sharing a screen? I don't think I am.
No, so if you mouse over the bottom of the screen, there should be a share button that's green, and you click on that.
Oh, sorry, I was doing it from my Zoom app. That was the wrong thing to do. All right. Do you guys see a screen?
Yes, we do.
Okay. All right, so I do think that thematically it works very well to go from Josh's talk to this one, because it's about what a virus chooses to do, not in a philosophical human choice way, but genetically, what are the choices viruses have? And a virus need not, come on. A virus, if it's replicating in a host, is already doing a pretty good job. It's making children. It can have a pretty good life. If there are a lot of hosts around, there's no need for a virus to think about having an expanded host range. It wouldn't be relevant in its life. However, when viruses do think of changing their host range, sometimes they spill over into humans, and then they become headline-grabbing viruses. A lot of what we know about host range evolution comes from human infecting viruses because we've studied them intensively. Intriguingly, most emergent viruses in humans need a host range mutation, but generally only about 1 point mutation to be able to infect new hosts. The human infecting viruses I'm showing here are RNA-based. This is not a fluke. RNA viruses are the most emergent pathogens in humans, in part because RNA viruses have high mutation rates. And sometimes those mutations will have an effect on host range. So I'm showing you a figure here that has on the y-axis mutation rate in per site in the genome per genomic replication, and along the x-axis genome size in log of kilobases. And I have stock art representing different kinds of organisms: a mouse for mammals, bacteria, a phage for big double-stranded DNA viruses, some of Arvin's presented beautifully on the small single-stranded DNA viruses here. RNA viruses do have a range. That's why we have a lengthy Ebola showing a range of RNA virus mutation rates. And up here we have a viroid, which is not viral, so we're not going to talk about it today. RNA viruses have high mutation rates. Sometimes those mutations are going to infect host range. What are the advantages of having mutations that affect host range in your population? What are the constraints of having a host range mutation? I've been studying this in a model RNA virus system, but a number of groups have studied this even in some human infecting systems. Generally, host range has been studied in the modern era in RNA viruses more than in DNA viruses. There's an obvious relationship between host range in viruses, which is a good proxy for the resource niche breadth of viruses, and how many virus progeny that we expect viruses to make in each given host. I'm not going quite as detailed back into the mathematical literature as Josh, but this model that a jack of all trades is a master of none has been articulated in the ecological literature, including mathematically since the 60s. That basically, if you are a virus whose niche is being illustrated by the red line, you're necessarily, we're going to call you a specialist because you infect a narrow range of hosts and you're better at infecting those hosts than the pink virus, whose we're calling the generalist. These are relative terms. You can't possibly call something a specialist or generalist without having another one to compare it to. There's no objective definition for specialism or generalism. Mathematically, people have models that predict that as your host range increases, as your niche breadth increases, you necessarily must get worse at fitness because the models assume that there's a similar amount of fitness under each curve in total. My pop culture metaphor for the jack of all trades is a master of none is aging. If anyone has suggestions for a more updated 2000 and teens or beyond metaphor, please put them in chat. What I tend to think about it is if you are specializing on a very specific kind of prey, you can be very, very good at catching it and keeping your narrow focus in the short time period that that prey may be around. But a generalist isn't necessarily tracking down one particular way of getting points. The generalist is aware that there are lots of ways to get points in the Pac-Man game. And so they're not going to necessarily catch as many ghosts, but they're going to get perhaps more points by diversifying their targets. In viruses, as I had said verbally before, the specialist we expect has more offspring from each infected host. When they get into a host, they're more productive. They get more fitness out of it. The pink generalist, we say it has a different set of advantages. it doesn't have to wait until it finds a suitable host. It can have reduced search time. It can come across lots of different hosts and have offspring in them, but we expect it not to have as many offspring in each host. I'm going to talk about 3 stories today. The first is a blast from the past, part of my thesis work, which actually looked at this in an RNA experimental system. Does a larger host range in and of itself reduce the fitness on the usual host? Do we see that just the acquisition of a host range mutation brings your fitness down and puts you on the generalist curve compared to the specialist curve? So the system I work with is the RNA phage Phy6, which is a fantastic model system in experimental evolution. It's incredibly popular because even though it is a bacteriophage, it looks like a eukaryotic virus. It has a lipid coat. Only Phy6 and its relatives in family Cystoviridae have a lipid coat. Only cystoviruses have a segmented genome. There is a 3 tripartite segmented genome. Segmentation was thought to evolve to deal with eukaryotic cellular machinery and their preferences, so it doesn't make a lot of sense that a tripartite genome would have evolved in prokaryotic viruses. And in terms of phylogenetic evidence, the RNA-dependent RNA polymerase, which is the only shared gene across all RNA viruses, when you do the phylogenetics of that gene, Phy6 groups with eukaryote infecting reoviruses and not with other RNA phages. It's got a pretty small genome, and it has a high mutation rate compared to the double-stranded DNA viruses that were being envisioned in Josh's talk. But because it's a double-stranded RNA virus, perhaps, it has the lowest measured mutation rate amongst RNA viruses. Phy6 is a phage. It infects Pseudomonas bacteria. I have a phylogeny here of the very broad Pseudomonas phylogeny outgroup rooted with E. coli. There are roughly 5 fairly distinct groups that, and I've given you a characteristic species name to represent each of them. Phy6's normal hosts are found in the syringe clade. It was isolated as a potential biocontrol agent for P. syringae pathomar phaseolicola, the cause of halo beam blight. You can easily get Phy6 to infect other P. syringae pathomars when they have host range mutations. And there's one other known alternative host you can use for Phy6. It is called Pseudomonas pseudoalkaligenes ERA. That stands for East River Isolate A. It was dredged up from the East River in the 1970s by molecular biologists working on Phy6, and we don't know much about its ecology. So a long time ago, I isolated host range mutants on 2 P. syringae pathomars, atrophosians and tomato, and on ERA, and I sequenced using Sanger sequencing to find that each one of my mutants had one of 9 different mutations in the same protein in Phy6, this P3 host attachment protein. So it was already a target that we would have expected and had been known for 30 years was a place where you could get host range mutants for Phy6. Of the 9 distinct mutations I found, 3 of them enabled Phy6 to infect atrophosians and ERA, 5 allowed it to infect tomato and ERA, and 1 allowed it to infect ERA alone. I then measured their fitness on the original host, P. syringae pathomar phaseolicola, to see if carrying these mutations already led to a fitness cost. And I measured fitnesses as a 24-hour paired growth assay, where the wild-type virus and a host range mutant could grow side by side, and we could see which one of them left more descendants after that much time. And in this figure, the y-axis is the fitness on the original host, Pseudomonas syringae pathomar phaseolicola. The fitness of the wild-type ancestor is shown by the dotted line at 1. Across the x-axis, you have the 9 different mutations in P3 that I found in that study. The color coding tells you what the host range of the descendant viruses are: red for infecting tomato and ERA, blue for atrophosians and ERA, black for ERA. What should be clear from here is that most of the time, but not all of the time, we see that there is an immediate loss in fitness on the original host just for carrying a host range mutation. There's been no ecological history. There's been no time for a lot of natural selection to occur in a novel host, but just having that flexibility in being able to attach and infect a couple of different hosts already is giving you an immeasurable fitness cost on your original host. 2 of our 9 mutations did not have a statistically detectable cost on the original host. As other people have done similar studies in other systems, including human viruses, we see the same pattern. Most of the time, we see a cost, an explicit antagonistic pleiotropy. but there seem to be a couple of ways that viruses can do this where we don't detect a cost in the lab of having carrying around an extra ability to have a wider host range. I'm going to take 2 of these particular host range mutations further into the next study. One from the N-terminal end of the P3 protein, one from closer to the C-terminal end. EHE and G515S is what I will refer to them as. So now we know that having a host range mutation in and of itself can cause a fitness effect. But how does having a host range mutation affect a virus's ability to jump into another novel host? How does it affect serial host shifting? So we have 2 mutants that infect our Phylosphere, tomato, and ERA, a wild type that only infects the original host Phylosphere. And we studied them in a couple of different ways to measure the mutational spectrum of how each of these 3 strains can get into a third host, Atrophacians. The way I'm going to show you results from is from Illumina sequencing. We grew up a high titer lysate of the virus on the last host it had experienced, so either the original host or, in this case, host tomato in those 2 strains. That was our before population. We then took a sample of that, put the rest of it onto a plate of Atrophacians and let it grow, and whatever plaques grew up, we harvested them and isolated their genomes. We now had 2 populations of viruses before and after exposure to Atrophacians, and we threw that onto Illumina MySeq. And the results we got are shown here. On the y-axis, I'm showing you the change in Shannon entropy. So what I'm showing you is the amount of polymorphism at the different sites, in this case in the M segment. This is representing 4,000-ish bases in the M segment. The after exposure to Atrophacians minus prior to exposure to Atrophacians. So anything you're seeing above the 0 line, which is represented by most of the genome, those are mutations that were overrepresented in the after Atrophacians exposure rather than before. I've given you a bit of shading to show you where the P3 host attachment protein is on the medium segment. The first thing you should see is that we're not seeing a lot of positive changes in Shannon entropy outside of the P3 region. We're seeing a lot of the host range mutations are really happening there. Also, you should be able to tell that there's more diverse sites associated with enrichment for the wild type population on the top, rather than the EHE or the G515S populations below. The wild type had more different ways to infect Atrophacians than the 2 strains that already had a host range mutation in their P3 gene. I'm showing you just the results from Illumina. We also did this with painstaking cloning and Sanger sequencing. And when we did that approach with groups of 50 individual mutants, we found that about 20% of the clones we pulled up from Phy6 wild type didn't have any mutations in their P3 gene. We were surprised by this. That was a really high result. Occasionally that's happened in Phy6 studies in the past, but it's been 1 out of 40, 2 out of 100. But it was closer to 1 out of 5 in this study. And the Illumina sequencing results gave us some perspective on what was happening there, because we got to see that there was similar enrichment in the small segment for Phy6 wild type, but not for Phy6 EHG or Phy6 G515S. None of the genes on the small segment had ever been associated with host range, and very intriguingly, the gene that has the most overrepresented sites both in our Illumina sequencing, and once we got these results, we went back and we did Sanger sequencing on our clones. We confirmed that they had single mutations, or in some case double mutations, in the P12 gene on the small segment. P12 is a protein that's not even in the fully assembled and articulated Phy6 variant. It's not part of the structure of the variant. So this is a really interesting result that there is this way that, so the Phy6 wild type can enhance its host range, possibly in helping it get out of that first infected Atrophacian cell. We don't have any, we're just speculating at this point, but those pathways appear to be closed off to the Phy6 strains that already have a host range mutation, EHG and G515S. All in all, wild type just has a lot of different ways to get into Atrophacians. Having a host range mutation is limiting you already. And this is clearly due to epistasis, an interaction amongst mutations. The mutations that are conferring the ability to infect Atrophacians for the wild type aren't necessarily working for those that already have a mutation in their P3 gene. We assume that this is a problem of P3 structural stability. that if you have a tweak that allows you to expand your host range and get into host tomato, another tweak, not as many of those secondary tweaks are tolerated, even though they would allow you to maybe get into another host. It may be reducing virion stability in some way, shape, or form. This is potentially a more generalizable thing beyond Phy6. While we see zoonotic spillover from a common source population into other hosts a lot, serial host shifting of a bird flu going into a horse and then going into a dog from the horse, which has happened for influenza, those kinds of sequential host shiftings are rarer. So epistatic interactions amongst host range mutations might be a generalizable problem for viruses. Again, this is a fairly short-term scale that we're examining the costs and benefits of host range mutations on. This is only a few generations, 10, 15 generations from the spontaneous host range mutation itself. We also have been giving our viral strains some time to evolve in different environments to see what the effects are on the evolutionary trajectory if you carry host range mutations. So this is a study that went for 30 overnight passages, so roughly 150 viral generations, with 4 treatments: the wild type Phy6 virus shown in teal, and the EAG virus shown in a pale beige color if it evolved just on the original host Phaseolicula. Wild type, of course, can only infect Phaseolicula, so we have wild type infecting Phaseolicula for 30 passages, EAG infecting Phaseolicula for 30 passages, or EAG alternating daily passage on the original host and a novel host, either tomato in red or ERA in black. So 4 different treatments that would allow us to compare the effects of genetic generalism by looking at the wild type evolving on Phaseolicula compared to EAG evolving on Phaseolicula, or the effect of ecological generalism. EAG, it has a host range mutation, but it's still seeing that original host, a constant host environment, compared to forcing it to see multiple hosts, either 2 in the P. syringae species or a P. syringae and a P. pseudoalkylogenes alternating days. The fitness results are shown on this slide. On the y-axis, we have relative fitness on the original host Phaseolicula. In this case, 1 does not mean the fitness of the wild type virus. It means the fitness of the common competitor we used to compare all of these strains to. The black box is the wild type fitness. Those teal are the 4 descended populations. Wild type evolved on the original host for 30 days. It's about the same fitness as it had when it started. The gray box is the original fitness of the EAG ancestor. The pale dots are after 30 days of evolution on Phaseolicula. Again, it was pretty decent at infecting Phaseolicula. We don't see a big change. In orange, we see the EAG populations that alternated with host tomato. In burgundy, the populations that alternated with host ERA. There are more data points for these latter 2 groups because we measure their fitness coming off of Phaseolicula and coming off of tomato or ERA, so day 29 and day 30, to make sure there wasn't a maternal host effect. When you compare between wild type and EAG grown on the same host, you don't see a lot of difference. There's not a big cost of carrying EAG longer term on the host Phaseolicula. They're both doing just fine. When you compare the different kinds of generalist environments that EAG could be in, we saw something pretty surprising, which is even though the EAG that was exposed to both Phaseolicula and tomato was seeing Phaseolicula every other day, some of the populations were losing fitness on the original host. There was so much selection on the novel host that this was apparently more important to the fitness of the overall multiple day fitness of these organisms, that we still are seeing fitness declining on the original host despite exposure to the original host. And I can show you some molecular evidence for that strong selection in this figure. Now I'm showing you the top 10 SNPs from each of 4 replicate populations that were evolved from the wild type on Phaseolicula, EAG on Phaseolicula, EAG alternating between Phaseolicula and tomato, EAG alternating between Phaseolicula and ERA. Light color pink indicates a low frequency, closer to 0. Dark purple indicates a high frequency, and darkest purple would be 1. And what you can see here is that of the top 10 SNPs, we don't see a lot of SNPs going to particularly high frequency in the populations evolving on Phaseolicula. Wild type and its isogenic EAG immediate offspring host range mutant were already pretty good at infecting Phaseolicula. We don't see a lot of change happening in 30 days. But the EHE populations that were experiencing a more generalist environment, we see some substantial selection. Some mutations are even being fixed because in the novel host, genetic diversity apparently is helping out these viruses get to a better fitness level. And so we see that there's a very different evolutionary scenario for the generalist viruses when they're in the different hosts. They are experiencing a selection that they weren't experiencing when they were just cruising along in their usual host. So perhaps unsatisfyingly, the answer to what are the benefits and detractions of having an expanded host range is, well, there's pluses and minuses. There absolutely are fitness costs and epistatic costs of host range mutations that can influence evolutionary trajectories. But whether or not these things are good or bad are going to depend on what kind of environment the virus lives in. If you're in an extremely homogeneous environment, let's imagine an industrial fermenter. There's no reason to have a lot of host range mutations. You want to kill the dominant bacteria, the only bacteria that they put in there to ferment the food substance they're interested in. If you're out in a more natural community with many, many different hosts around you, it would make a lot more sense to have some expanded host range mutations in your standing population genetic variation, even if they confer small levels of fitness effects on the host you more typically encounter. Most of the work I presented today was the thesis work of Lola Zhao, who's now a postdoc at Oxford Big Data Institute. She was helped by 2 undergrads, Dragos Stamate and Alvin Crespo, and Mancha Seth Pasricha, a postdoc in my group, was also contributed to the Mutational Spectrum Project. Thank you so much for listening. Thank you for the invitation, organizers, and I'm happy to take questions.
I actually have a question to start off.
Sure. Hi, Eric.
So if we're thinking about culturing viruses for the future, this idea that maybe to increase our ability to culture viruses, we want viruses that can infect many different types of hosts with a broad host range, right? So you're saying if we go along this line, that if we have a more simple environment, which selects for a certain type of metabolism maybe, that it will be harder to culture viruses from those type of environments?
I don't know if we know enough to answer one way or the other at this point. Even though we didn't see many high-frequency SNPs in our populations that survived on just the original host Phylosphere, they did have a higher diversity overall than our populations that were seeing a wider number of hosts, because selection purges diversity at linked sites. You know, we always forget this. You know, evolution's great. Natural selection's great, except it's getting rid of that lovely diversity that helps the next step that goes along. I'm not sure we have a science-based answer to that question yet.
Thank you.
Don, I had a question, sort of a more general one. Have you or anyone else looked at some of the more broad host range viruses? Because in this case, you're looking at these individual mutations in a nice, well-defined system. But if you think about it from some of the cucumber mosaic viruses, or those that have much broader host ranges, do you see the same kinds of trade-offs, fitness or otherwise? Or has anybody really done good experiments with that? I imagine that's hard to do.
We know that broad host range plant viruses see different plant hosts differently. And a lot of that's the work of Marilyn Rusnick's group. Santiago Lane has some work too, but Marilyn really characterized that the kinds of mutations that are favored in the different environments it's clearly different in different plant hosts. But in plant viruses, we don't see a lot of host-specific adaptation. So you see this when you're infecting a plant, but it doesn't seem to persist when you're looking at the between host evolution, even if you're keeping them in the same host for long periods of time. It's been a frustrating difference between the phage literature and the plant literature, is that there's much more host-specific adaptation that you can measure in the phage literature.
Mm-hmm. Is that possibly because of how they're being infected?
Yeah.
Since vector-borne or non-vector-borne?
And my postdoc also was whispering the exact same thing. Plant viruses are often vector-specific and host generalist, but these sorts of patterns occur even in wind-transmitted plants. viruses. So this isn't something that's restricted because you have to look a certain way to go with the vector between hosts. On a population level, even with the same host, we don't see as much host-specific adaptation. When people look at this question of broader host range in animals, they do shorthand it to arboviruses versus something that's mammal-specific, and that's not a great comparison either because, yeah.
Thank you. Any other questions? I'm curious, have you done similar experiments with single-stranded DNA viruses?
Our single-stranded DNA phage in the lab is 5x174, and it is pretty difficult to get onto alternative hosts. There's a very short list of alternative hosts for that particular virus. It doesn't have a massive host range mutation generation ability. It's frustrating. And I work with many people who work on the Gemini viruses, the biggest group of plant-infecting single-stranded DNA viruses, and the lack of host-specific adaptation is just bizarre.
Thanks. Yeah, of course we're looking at some of these strange single-stranded DNA viruses as well, and love to have some handles on it.
They're everywhere, and someone should develop one of the ones that infects fungi or some protist into a good working experimental system.
I agree.
I'll take an archaeal system if you can get that thing, the 25 kb one, to grow well.
I'd like to tell Mohiro about that. Arvind had a nice notice in the chat group about that plant virus evolution and ecology. That was a recent, Arvind, are you still around online here? That was a recent review, correct?
Yes, that is correct. So that kind of covers a lot of work from ecology to evolution, also looks at trade-off hypothesis in there, talks about insects playing a major role and how insects and plant viruses have potentially co-evolved, primarily because of the vector dynamics.
Yeah, thanks. It's I can say this because I'm not one of the authors. It's a great read, so highly recommend it.
But we're still at the point where they have to have special mixer sessions at ASV for the animal and plant virus people to talk to each other and try to figure out what's similar across our disciplines, and those rooms don't include the phage people yet, or the archaeovirus people.
The vast numbers of us.
I think if we take hands on this call, we'll be fine.
No. Which actually I think is a great transition. So in theory, in the schedule, I'm supposed to say something about closing remarks. I'd actually much prefer to open this up to really everyone to make comments. And unfortunately, I snuck out at the break, but it seems that there were some issues with seeing yourself and not seeing yourself as far as being bizarre for presentation-wise. Do we have any more feedback on that or any other questions for other people before we sort of wrap up for today and then pick up again tomorrow afternoon? Anybody at NAI Central have some comments, thoughts? I completely agree with the chats. We need to try and get a transcript for that. I think there's a lot of really cool stuff going on there. I just haven't been able to split my attention. I'm really bad at multitasking, so.
Well, I've been paying attention to the chats and answering a few things and asking a few things, so we will have those captured. We also would like to make sure that everyone from whom we have slides is willing to have us post them on the NAI website under this workshop. So I guess we'll ask anyone who presented who doesn't want that to happen to certainly email us right away. If not, we'll certainly confirm with you before we post them. if you need to take any proprietary or sensitive information out, or who knows what, if you need to do a cleanup on them. And after that, within a couple of days, we'll have those posted. Also, we've recorded this, so for any of you who joined us late or intermittently, you'll be able to catch the parts that you didn't, you weren't able to join us for. And then for anybody who wasn't present right at the beginning, one of the aspirations of this effort is to produce tangible product, and one is a journal article produced by you folks. I'll facilitate it as much as I can for the journal Astrobiology, and then a distillation of that for the upcoming planetary decadal white papers. So that's just to recap what I said in the beginning for those of you who weren't on. And with that, I think that's all I need to say, except I had a great time. And a lot of it went over my head.
Yeah, thanks. And any comments, if you don't feel like sharing them in a general chat, feel free to do a direct chat to any of us. As far as that's concerned, you can also find our email addresses, etc. I haven't been watching on Twitter. I'm sure we've completely blown up. Yeah, let's see. And thanks again, Penny, for pointing out the article and white papers. We will be reaching out to you. I guarantee that. That will happen. Keep an eye on your inboxes, and we will let you know about that. I will try and synthesize a little bit more of what I've heard today. As I was personally chatting with someone, even I'm a little overwhelmed at this point, so I need to sit back and synthesize some of these things, maybe re-listen to a couple of the presentations, and I'll try and fit some of that into my closing talk tomorrow as well.
One other point that I want to make, we've had a great turnout for this. The highest number of participants on at any one time was 87. And we probably have a few more of that in terms of, total coming and going. So that exceeds my expectations for how many people we could pull out of the woodwork who are interested in this, whether or not they happen to be experts. So I think that there's a lot of interest in the community, and that makes it worth having gone to the trouble of doing this, to try to get everybody to be talking to each other, including folks that may not consider themselves virologists. We discussed this at the beginning. There are many people who are not virologists, but who have probably things to contribute, and getting them also thinking along the lines of the role of viruses and other mobile genetic entities that are moving things around, I think is really worthwhile for the effort. And so I look forward to seeing everybody tomorrow afternoon, and maybe a few additional people that we didn't have who couldn't make it today. So thanks everybody for attending.
I wanted to throw one last bit in there. If you're not a virus specialist, or even if you are, you may have not understood some of the jargon that was discussed today. On our website, we have some helpful resources, but also feel free to email us with any questions. There's no bad question. If you want to know what is a lytic virus or something, please email us so we can help you out. That way, if you feel like it might help you be more engaging for tomorrow or might be useful for you, we're happy to help.
Yeah, all I can say is thank you. To everybody who's participated. This has been really, really wonderful, at least from my highly biased point of view. So thanks to everybody, and hopefully we'll see all of you, or at least hear from some of you tomorrow.
Great.
Thanks
Thanks everybody. Bye.
Thank you.